There is a silence in a forest that is not really silence at all. Stand among ancient oaks or towering redwoods and you begin to sense it, an almost imperceptible hum beneath the surface of things, a slow choreography of chemistry, physics, and memory that has been unfolding for hundreds of millions of years. Trees are the oldest large organisms on Earth, and in many ways the most profoundly misunderstood. We walk past them daily, sheltered beneath their canopies, breathing the oxygen they exhale, and yet we rarely pause to consider what they truly are. They are not passive backdrops to the drama of life. They are the stage, the actors, and frequently the directors. They communicate, compete, nurture, and sacrifice. They remember droughts and record history in their very bones. They have shaped the atmosphere, the soil, the rain, and the minds of every civilization that has ever flourished in their shade. This documentary is an invitation to look closer, to see the forest not as scenery, but as one of the most extraordinary, mysterious, and quietly magnificent systems the universe has ever assembled. Long before the first tree stood upon this earth, the land was a barren, wind-scoured expanse of rock and shallow water. It was approximately 385 million years ago, during the Devonian period, that evolution made one of its most consequential leaps: the emergence of woody plants capable of growing tall. The earliest tree-like organisms, such as Archaeopteryx, were not fully trees in the modern sense. They bore fern-like fronds and reproduced through spores rather than seeds, but they possessed something revolutionary: lignin. This complex organic polymer, still found in every woody stem today, allowed plants to build rigid, self-supporting structures that could reach toward the sun far above their competitors. The evolution of trees fundamentally transformed the planet. Their roots broke down rock and produced soil. Their canopies altered rainfall patterns. Their mass burial over geological time created the coal seams we now extract for energy. In this sense, the Industrial Revolution was, in a remarkable way, powered by the ancient forests of the Carboniferous, by trees that fell and compressed over 300 million years, waiting in geological patience for a species clever enough, or reckless enough, to release them. To understand a tree, one must begin where it begins, underground. The root system of a mature tree is a marvel of engineering that most of us will never see, hidden beneath our feet in a dark and intricate world. Far from being simple anchors, roots are dynamic organs engaged in ceaseless chemical negotiation with the soil. A single mature oak may possess a root network spanning hundreds of square meters, extending outward well beyond the drip line of its canopy and probing downward through rock fractures in search of water and minerals. The fine root hairs at the growing tips of roots are among the most sensitive biological structures in nature, capable of detecting minute chemical gradients, moisture differentials, and even electrical signals. Roots actively choose their direction of growth through a process called tropism, bending toward water, away from toxins, and downward in response to gravity. They exude sugars and acids into the soil to dissolve minerals and attract beneficial microbes, essentially farming the organisms around them. This relationship between root and soil is not exploitation but coevolution, a partnership refined over hundreds of millions of years into something approaching mutual dependency. Rising from the ground, the trunk of a tree is an architectural achievement of surpassing elegance. Its primary purpose is mechanical: to hold the canopy aloft where light is available and to serve as a conduit between the root system and the leaves. But the trunk is far more than a wooden pillar. At its core lies the heartwood, dense, often richly colored, no longer biologically active but structurally essential, composed of dead cells impregnated with resins, tannins, and minerals that make this inner wood remarkably resistant to decay. Surrounding the heartwood is the sapwood, or xylem, alive and actively transporting water and dissolved nutrients upward from the roots. Between the xylem and the outer bark lies a tissue of extraordinary importance: the cambium, a thin sheet of dividing cells just a few layers thick that generates all new wood and bark. It is the cambium that produces the annual growth rings visible when a tree is cross-sectioned. The outer bark, meanwhile, is the tree's armored skin, a layered shield against insects, pathogens, fire, and physical damage, composed of cork cells that are largely dead, their walls saturated with suberin, a waxy polymer that makes them nearly waterproof. Among the most astonishing feats performed by trees is the daily transportation of enormous quantities of water from root to leaf against the force of gravity. A large oak on a summer day may transpire hundreds of liters of water through its leaves, and every drop of that water must be drawn up from the soil through vessels in the wood, sometimes ascending 30 or 40 meters without the assistance of any pump. The mechanism by which this happens is both elegant and improbable. Water molecules are polar, meaning they form hydrogen bonds with one another, producing a property called cohesion. They essentially cling together in continuous chains. As water evaporates from the leaf surface through tiny pores called stomata, it pulls on the chain below it, generating a tension that propagates all the way down through the xylem vessels to the roots. This is the cohesion tension theory of water transport, and it means that the water column inside a tree is perpetually under negative pressure, technically in a state of metastable tension that would shatter if a bubble formed within it. Trees manage this extraordinary physical state with astonishing reliability across decades and centuries of continuous operation, in conditions ranging from drought to flood, heat to ice. While the xylem carries water upward from root to leaf, the phloem conducts an equally remarkable flow in the opposite direction, and sometimes simultaneously transporting dissolved sugars, hormones, and signaling molecules from leaves to every other part of the tree. The phloem is composed of sieve tube elements, living cells aligned end-to-end with perforated walls that allow fluid to pass between them. Unlike xylem transport, which is driven by evaporation and physical tension, phloem transport operates by a pressure gradient created by differences in sugar concentration. In the leaves, where photosynthesis generates sugars, these molecules are actively loaded into the phloem, raising the osmotic pressure and drawing water in from nearby xylem vessels. At the roots, stems, and fruits, which consume or store sugars, they are unloaded, reducing pressure there. This difference in pressure drives a continuous flow of phloem sap from source to sink, in a mechanism known as pressure flow or the Munch hypothesis. The elegance of this system lies in its self-regulating nature. The greater the photosynthetic activity in a leaf, the stronger the pressure gradient and the faster the flow. The tree, in effect, continuously calibrates its internal economy without any central controller. The leaf is perhaps the single most consequential structure in the history of life on Earth. In its thin, flat, unremarkable-seeming blade lies the machinery for photosynthesis, the process by which light energy from a star 93 million miles away is captured and converted into the chemical bonds of sugar, the foundational currency of almost all life. A single leaf is an engineering marvel. Its shape maximizes light capture while minimizing the distance water must travel to evaporating surfaces. Its internal architecture, layers of palisade cells packed with chloroplasts, below which lie the loosely arranged spongy mesophyll cells threaded with air spaces, optimizes both light absorption and gas exchange. The stomata, those tiny pores on the leaf surface controlled by pairs of guard cells, represent one of evolution's most sophisticated regulatory mechanisms, opening to admit carbon dioxide and closing to prevent water loss with a responsiveness that adjusts moment by moment to light intensity, temperature, humidity, and the tree's water status. Some trees possess tens of millions of leaves, each one processing solar energy, exchanging gases with the atmosphere, and communicating with the rest of the organism through the vascular system below, a distributed solar array of breathtaking complexity. The process of photosynthesis, when examined closely, is so intricate and improbable that it continues to astonish the scientists who study it. It occurs in two broad stages. The first, the light-dependent reactions, takes place in the thylakoid membranes of the chloroplast, where chlorophyll molecules absorb photons of light and use that energy to split water molecules, releasing oxygen as a byproduct and harvesting electrons and protons to produce energy-carrying molecules called ATP and NADH. This is the source of virtually all the oxygen in Earth's atmosphere. The second stage, the Calvin cycle, occurs in the stroma of the chloroplast and uses the energy from ATP and NADPH to fix carbon dioxide from the air into 3 carbon molecules, which are then used to build glucose and other sugars. What makes this process so remarkable is not merely its chemistry, which is extraordinary in its own right, but its sheer efficiency under natural conditions. Trees manage to perform these reactions reliably in environments ranging from Arctic tundra to tropical rainforest, adjusting the chemistry of their chloroplasts in response to temperature, light, and available nutrients in ways that biochemists are only beginning to fully understand. In the cross-section of a felled tree, time becomes visible. Each ring of wood represents 1 year of growth. A pale band of large-celled spring wood formed during the flush of early-season growth, bordered by a darker band of denser summer wood produced as growth slows toward autumn. The science of reading these rings, known as dendrochronology, was systematized in the early 20th century by the astronomer Andrew Ellicott Douglas, who originally hoped to find correlations between sunspot cycles and tree growth. What he discovered instead was far more profound: that tree rings are extraordinarily sensitive archives of environmental history, recording not just time but climate. A wide ring indicates a favorable year, warm, wet, abundant. A narrow ring speaks of drought, late frost, volcanic ash clouds, or insect defoliation. Because trees of the same species in the same region tend to grow wide and narrow rings in the same years, their ring patterns can be matched and cross-dated, a technique called cross-dating, allowing scientists to build continuous chronologies that extend thousands of years into the past. These chronologies have been used to calibrate radiocarbon dating, reconstruct ancient climates, and date historic structures from the wood preserved within them. The power of dendrochronology to illuminate the past grows steadily as researchers extend ring chronologies further back in time. Using preserved wood from ancient buildings, peat bogs, lake sediments, and subfossil logs, trees that died centuries or millennia ago and were preserved in anaerobic conditions, scientists have built regional ring chronologies extending back more than 10,000 years in some parts of Europe. These records have revealed a startling richness of climatic detail. The great volcanic eruption of Samalas in Indonesia in 1257, one of the largest in recorded history, is written in tree rings around the globe as a period of dramatically narrowed growth. The following summers were cold and dark from volcanic aerosols. The medieval warm period and the Little Ice Age, long debated by climatologists, are clearly visible in ring data from across the northern hemisphere. The prolonged drought that may have contributed to the collapse of the Maya civilization, the societal disruptions caused by volcanic cooling events in the late Roman Empire, all of these events leave their signatures in wood that was growing silently in forests while empires rose and fell. Trees, it turns out, are the most patient and meticulous historians our planet has produced. High in the White Mountains of eastern California, at an elevation above 3,000 meters, where the soil is poor and wind-blasted and snow lingers well into summer, grow some of the oldest living organisms on Earth. The Great Basin bristlecone pine, Pinus longiva, thrives in conditions that would kill most trees, and it is precisely this adversity that grants it extraordinary longevity. Stress limits growth, and slower growth produces denser, more resin-saturated wood that resists decay, disease, and insect attack. Among these ancient sentinels lives a tree known as Methuselah, its location kept secret by the United States Forest Service to protect it from vandalism. Methuselah is approximately 5,000 years old, making it the oldest verified living non-clonal organism on Earth. It was already ancient when the pyramids at Giza were under construction, when Stonehenge was being assembled, when Homer composed the Iliad. Its gnarled, wind-sculpted form gives no outward indication of its extraordinary age. It is relatively modest in stature, its slow growth producing wood so dense and resin-rich that it is virtually immune to the fungi and beetles that eventually claim younger, faster-growing trees. In Methuselah, we encounter time made biological, the living past. In the Fish Lake National Forest of south-central Utah stands what may be the most remarkable organism on Earth. Not a single tree, but a colony of approximately 47,000 quaking aspen stems, all genetically identical, all sharing a single, massive root system that spreads across more than 43 hectares. This organism, known as pando, which means ice spread in Latin, is estimated to weigh approximately 6 million kilograms, making it possibly the heaviest known organism on the planet. Its age is estimated between 80,000 and 1 million years, though the upper bound remains contested, making it potentially one of the oldest organisms ever to have lived on Earth. What appears to be a grove of individual trees is in reality a single superorganism, each pale-trunked stem merely a shoot arising from the subterranean root network. When the aspens tremble in the breeze, sending their silver-green leaves shimmering in unison, they do so as a collective, connected entity. Pando challenges our conventional understanding of what constitutes an individual in biology. It also faces contemporary threats: overgrazing by deer and elk, drought intensified by climate change, and fungal disease are suppressing the regeneration of new stems, and this ancient giant is, for the first time in its extraordinary existence, in measurable decline. In a remote and undisclosed location within California's Redwood National Park stands Hyperion, the tallest known living tree on Earth. Discovered in 2006 by naturalists Chris Atkins and Michael Taylor, Hyperion is a coast redwood, Sequoia sempervirens, measuring 115.92 meters in height at its last verified measurement. To stand at its base and crane your neck upward is to experience a kind of architectural vertigo. The trunk rises for over 100 meters before the canopy begins, a column of rust-red bark 20 or more centimeters thick disappearing into the green-shadowed heights above. Coast redwoods are the tallest tree species in the world, and their extraordinary height is sustained by a combination of abundant moisture from coastal fogs, mild temperatures, deep fertile soils, and freedom from the insects, fungi, and fires that limit the height of trees in other climates. Tannins in the wood make it profoundly resistant to both fire and biological decay, and there are living redwoods that were already mature trees before the birth of Christ. Hyperion itself is believed to be somewhere between 600 and 800 years old. Its precise location is deliberately withheld to prevent the compaction of soil around its roots by curious visitors, a fragility hidden within an apparent titan. For most of scientific history, trees were understood as fundamentally solitary competitors, each individual striving against its neighbors for light, water, and nutrients, the forest a battlefield where only the strongest survived. This view began to change dramatically in the 1990s, when the forest ecologist Suzanne Simard, working in the forests of British Columbia, conducted a series of elegant experiments that would rewrite our understanding of how trees interact. By tagging carbon dioxide with radioactive isotopes and tracking where those carbon atoms traveled after being fixed by photosynthesis, Simard discovered something extraordinary. Carbon was flowing underground between trees of different species, transferred through their shared fungal partners. She had found evidence for what would eventually be called the mycorrhizal network, colloquially known as the wood wide web, a subterranean web of fungal filaments connecting the root systems of trees across large areas of forest floor. The implications were profound. Trees were not only competing but actively exchanging nutrients, and the flow of resources was not random. It appeared to move preferentially from larger, more productive trees to smaller, shaded seedlings. The forest, it seemed, was operating as something more like an integrated community than a collection of warring individuals, though the full picture, as always in science, would prove considerably more complex. The architects of the wood wide web are the mycorrhizal fungi, a diverse assemblage of fungal species whose thread-like hyphae, individually invisible to the naked eye, collectively form networks of extraordinary extent and complexity. The word mycorrhiza comes from the Greek for fungus and root, and the relationship it describes is ancient, having evolved at least 400 million years ago, around the time the first plants colonized the land. In exchange for sugars produced by the tree through photosynthesis, mycorrhizal fungi extend the effective absorptive surface of the root system by orders of magnitude, reaching into soil pores too fine for roots to penetrate and accessing water and mineral nutrients, particularly phosphorus and nitrogen, that would otherwise be unavailable to the tree. This exchange is the foundation of the relationship, but the network that forms when many trees connect to the same fungal partner becomes something more: a conduit for the transfer of carbon, water, nutrients, and signaling molecules between trees. Different fungal species connect with different tree hosts, and a single tree may be simultaneously linked to dozens of different fungal species. The network is not a simple web but a layered, dynamic, and astonishingly responsive system that adjusts its connections over time in response to the needs and status of its participants. Within the wood wide web, not all nodes are equal. Certain large, old trees, particularly those whose canopies dominate the forest and whose root systems are the most extensively networked, play a disproportionate role in the fungal web, connecting to a greater number of neighboring trees and transferring larger quantities of resources. Suzanne Simard termed these individuals mother trees, a phrase that, while scientifically contentious in some circles, captures something genuinely important about their ecological function. Her research suggested that when mother trees are removed from a forest, as frequently happens in logging operations that selectively harvest the largest, most commercially valuable specimens, the surrounding seedlings and younger trees suffer measurably, losing access to the carbon subsidies and fungal connections that the mother tree had been providing. Even more provocatively, Simard's later work suggested that mother trees may preferentially allocate resources to their own offspring, seedlings genetically related to themselves, raising the extraordinary possibility that trees possess some form of kin recognition mediated through the fungal network. This claim remains actively debated among forest ecologists, and the mechanisms by which it might operate are not fully understood, but the evidence that tree identity and relatedness influence resource flow in mycorrhizal networks is accumulating and has fundamentally altered how we conceptualize forest ecology. Trees do not communicate only through underground networks. They speak also through the air. When a tree is attacked by herbivorous insects, its leaves being chewed by caterpillars or its bark bored by beetles, it responds with a rapid and sophisticated chemical cascade. Within minutes to hours, the damaged tissue begins producing volatile organic compounds, complex molecules including terpenes, methyl jasmonate, ethylene, and green leaf volatiles that diffuse outward through the air in all directions. Neighboring trees of the same species, detecting these chemical signals through their own leaf surfaces and stomata, respond by upregulating their own defenses before the attackers have reached them, producing more tannins, more toxic alkaloids, thickening cell walls, and increasing the production of chemicals distasteful or harmful to the anticipated herbivores. This phenomenon, sometimes called talking trees, was controversial when first proposed by ecologists David Rhodes and Ian Baldwin in the 1980s, and considerable scientific debate has surrounded it ever since. Some researchers question whether the response in neighboring trees is a genuine communication, an adaptation benefiting the signaling tree, or simply an incidental reaction to damage-related chemicals in the air. But whatever the evolutionary mechanism, the ecological reality of chemically mediated, inter-individual defense priming among trees is now well established. The chemical arsenal available to a tree under attack is vast, diverse, and frequently remarkable in its ingenuity. Tannins, polyphenolic compounds that bind to proteins and make plant tissue difficult to digest and unpalatable, are among the most widespread defensive chemicals in trees. Oaks are famously rich in tannins, and research has shown that oak tannin production increases measurably following herbivore attack. Many African acacia trees, famously, increase their leaf tannin concentrations so rapidly when giraffes begin to browse them that the giraffes move downwind to find trees not yet warned by the volatile compounds released by the grazed individual, a behavior long noted by wildlife observers and now understood in chemical terms. Beyond tannins, trees produce a remarkable pharmacopoeia of secondary metabolites whose primary function is defense: alkaloids that interfere with the nervous systems of insects, terpenoids that deter fungal colonization, latex and resin systems that physically trap and suffocate boring insects, phytoectosones that disrupt the molting of insect larvae, and compounds that mimic insect hormones, disrupting reproduction. Many of these compounds have proven medically valuable to humans. Taxol, derived from Pacific yew bark, is an important cancer chemotherapy agent. Aspirin's precursor was extracted from willow bark. Quinine from cinchona bark has saved millions from malaria. Trees shape not only the atmosphere above them but the soil chemistry beneath, deploying another dimension of chemical warfare through compounds known as allelochemicals, substances released from roots, fallen leaves, and decomposing bark that inhibit the germination and growth of competing plants. Allelopathy, as this phenomenon is called, is one of the most ancient and widespread forms of chemical competition in the plant kingdom, and trees have evolved some of its most potent examples. Black walnut trees, Juglans nigra, produce juglone, a quinone compound that is toxic to a wide range of plants and that persists in the soil around a walnut long after the tree itself is gone, explaining the curious barrenness of soil beneath and near walnut groves. Eucalyptus trees, native to Australia but invasive in many parts of the world, produce allelochemicals that leach from their fallen leaves and create notoriously plant-hostile soils, one mechanism behind the sparse understories characteristic of eucalyptus stands. Even more intriguingly, research has suggested that some tree root exudates not only inhibit competitors but selectively stimulate beneficial fungi and bacteria, essentially gardening the microbial community in the rhizosphere to create conditions favorable to the tree itself. The tree's influence, it becomes clear, extends far beyond its visible boundaries. Not all tree defenses are chemical. Evolution has furnished trees with a remarkable array of physical armaments and structural adaptations that deter predators, resist environmental extremes, and protect vital tissues from damage. Thorns and spines, produced by many species from hawthorns to honey locusts to Osage orange, represent the most visually obvious physical defenses, though their effectiveness varies significantly with the size and type of herbivore. More subtle but equally effective is the production of latex, a milky fluid under pressure in specialized internal vessels called laticiphers, which bleeds from wounds and rapidly coagulates on exposure to air, sealing the injury and trapping small insects that attempt to bore into the damaged tissue. The rubber tree, Hevea brasiliensis, produces latex in such quantities that it has given us the foundation of the rubber industry, but latex systems evolved purely as a defense against wood-boring beetles and other herbivores. Perhaps the most architecturally dramatic defensive structure in trees is the development of burls, gnarled woody growths produced in response to bacterial infections, fungal attacks, or insect infestations. By walling off an infected area with dense, irregularly grained wood, the tree prevents the pathogen from spreading while simultaneously creating some of the most beautiful and sought-after figured wood known to woodworkers. Fire is among the most ancient and powerful selective forces in forest ecosystems, and trees have evolved a breathtaking variety of strategies for not merely surviving fire but actively exploiting it. The coast redwood and giant sequoia possess bark up to 30 centimeters thick in mature individuals, bark so densely packed with tannins and so poor in flammable resins that surface fires scar but rarely penetrate it. Giant sequoias have taken this adaptation further still. Their cones remain sealed on the tree for 20 years or more, opening and releasing seeds only when the heat of a passing fire melts the resinous seal, ensuring that seeds fall into the ash-cleared, competitor-free soil immediately after a burn. The lodgepole pine exhibits similar serotiny, fire-responsive cone opening, and in the great coniferous forests of the American West, lodgepole and sequoia are ecological partners of fire, shaped by it and in some respects dependent upon it for regeneration. The cork oak of Mediterranean Europe produces bark so thick and chemically resistant that individual trees survive repeated harvesting by humans as well as wildfire, regenerating their bark layer with remarkable rapidity. Some Australian eucalyptus species possess lignotubers, woody swellings at their base packed with dormant buds, from which they resprout vigorously after fire destroys everything above ground. The reproductive strategies of trees represent some of evolution's most extravagant experiments in chemistry, structure, and ecological partnership. Angiosperms, the flowering trees that dominate most of the world's forests, have evolved flowers of extraordinary diversity, each one an advertisement finely tuned to attract specific pollinators. The flowers of cherry and apple trees offer nectar and pollen to bees in exchange for cross-pollination. Magnolia flowers, among the most ancient of flowering tree blossoms, are visited by beetles, which ate pollen before bees existed. Wind-pollinated trees such as oaks, birches, and alders produce enormous quantities of pollen, a single birch catkin releasing millions of grains, in a statistical strategy that compensates for the inefficiency of random dispersal through the air. The timing of flowering is exquisitely calibrated to environmental cues, day length, temperature accumulation above a threshold known as growing degree days, and in some species even the timing of rainfall. Climate change is disrupting these phenological calendars, causing mismatches between the flowering of trees and the emergence of their pollinators, a phenomenon of growing ecological concern as warming progresses faster than evolutionary adaptation can follow. The quiet explosion of spring blossom across a forest is, beneath its beauty, a precisely orchestrated chemical and ecological contract. A seed that falls directly beneath its parent tree faces a formidable challenge. It must compete for light and nutrients with the very individual that produced it, in soil already depleted by established roots, in shade cast by a mature canopy. Evolution has therefore furnished trees with the remarkable diversity of mechanisms for dispersing their seeds as far as possible from the parent. The maple employs aerodynamics. Its seeds are encased in paired samaras, papery wings that cause the seed to spin as it falls, slowing its descent and allowing wind to carry it considerable distances. The coconut palm trusts to the sea. Its buoyant, waterproof husk enables seeds to float for months and across ocean currents to colonize distant shores. The cherry, apple, and oak, among countless others, have contracted with animals. Their fruits and nuts are nutritionally valuable enough that birds, squirrels, bears, and jays carry them considerable distances, often caching them in locations ideal for germination and forgetting some fraction of their stores. The acorn caching behavior of jays in particular has been identified as a major driver of post-glacial oak forest expansion, jays effectively planting forests. Some trees exploit explosive mechanics. The sandbox tree of the Caribbean propels its seeds at velocities over 70 meters per second when its fruit dehisces, scattering them up to 45 meters from the parent. Not all mycorrhizal partnerships are alike. Broadly, two major types have evolved, each with distinct anatomy, ecology, and evolutionary history. Ectomycorrhizal fungi, the dominant partners of most temperate forest trees including oaks, beeches, pines, and birches, wrap around and between the cells of the root tip, forming a sheath of fungal tissue called the Hardig net without penetrating the root cells themselves. Endomycorrhizal fungi, by contrast, and particularly the arbuscular mycorrhizal fungi that partner with approximately 80% of terrestrial plant species, actually penetrate root cell walls and form branching tree-like structures within the cells called arbuscules, through which nutrient exchange occurs. Ectomycorrhizal partnerships are most common in nutrient-poor soils and in seasonally cold climates. Arbuscular mycorrhizal partnerships dominate in tropical forests and grasslands. The distinction matters enormously for forest ecology. Ectomycorrhizal fungi are much more efficient at mobilizing organic nitrogen and phosphorus from decomposing organic matter, giving ectomycorrhizal forests a distinct competitive advantage in nutrient-poor boreal and temperate environments. The diversity of fungal species involved in these networks is staggering. A single hectare of temperate forest may harbor over 1,000 species of mycorrhizal fungi, each with distinct host preferences, nutrient specializations, and environmental tolerances. In 1982, the Japanese Ministry of Agriculture, Forestry, and Fisheries coined the term Shinrin-yoku, forest bathing, to describe the practice of spending mindful, unhurried time in forested environments as a public health strategy. The concept, rooted in centuries of Japanese Shinto reverence for natural spaces, was given scientific framework in subsequent decades as researchers at institutions including Nippon Medical School began publishing rigorous studies on the physiological and psychological effects of time spent in forests. The findings were striking. Controlled studies comparing time spent in forested environments versus urban environments found that forest exposure measurably reduced levels of cortisol, the primary stress hormone, lowered blood pressure and heart rate, increased the activity of natural killer cells in the immune system, the frontline cells that identify and destroy cancerous and virus-infected cells, and reduced activity in the prefrontal cortex region associated with rumination and repetitive negative thought. These effects persisted for hours and in some studies for days after the forest visit. Japanese researchers formally designated specific forest trails as therapy roads, and the practice of forest bathing was incorporated into preventive medicine recommendations in Japan. What had been intuited by poets, contemplatives, and healers across millennia was at last being mapped in the language of biochemistry. The measurable health benefits of time spent in forests have a partially chemical explanation, one that connects the immune-enhancing effects of Shinrin-yoku to the very defense mechanisms trees employ against their own pathogens. When trees produce volatile organic compounds to deter bacteria, fungi, and insects, those same compounds drift through the air of the forest and are inhaled by any organisms breathing within that space. The compounds responsible for much of what we smell in forests, particularly the distinctive, resinous scent of pine and cypress forests, are known as phytoncides, a term coined by the Russian biochemist Boris Tokhin in the 1920s. Chief among these are alpha-pinene and beta-pinene, monoterpene compounds released in greatest quantities by conifers. Research by the Japanese immunologist Qing Li found that exposure to phytoncides in forested environments measurably increased the number and activity of natural killer cells in human subjects, and that this immune-boosting effect could even be replicated, to some degree, by exposing subjects to diffused phytoncide compounds in urban hotel rooms. The forest, in this sense, medicates all who walk through it, a pharmaceutical dispensed as perfume, shared freely with every breath, an ancient chemical conversation to which humans are uninvited but nonetheless chemically responsive guests. Across virtually every human civilization that developed in regions forested enough to sustain significant tree growth, the tree has occupied a position of profound sacred importance. The recurrence of the world tree motif, a cosmic tree whose roots reach into the underworld and whose branches support the sky, across cultures that had no known contact with one another, speaks to something deep in the human relationship with these organisms. The Norse Yggdrasil, an immense ash tree at the center of 9 worlds, connecting Asgard and Midgard and Hel, nourished by 3 great wells at its roots, inhabited by an eagle, a serpent, and a squirrel that carries messages between them, is perhaps the most elaborately developed world tree mythology. But the same essential image appears in the Mayan Huacachan, the pillar tree of the center connecting earth, sky, and underworld. It appears in Hinduism as the cosmic Ashvatha tree, the sacred fig beneath which the universe grows and perishes. It appears in Siberian shamanic traditions where the shaman climbs a spirit tree to travel between worlds. The biological reality, that trees literally connect underground and atmospheric worlds, that they are inhabited by countless species, that their roots and crowns genuinely span different ecological domains, gives these mythologies a resonance that is more than metaphorical. Beyond the world tree concept, specific tree species have been elevated to sacred status in cultures around the world, often in ways that reflect genuine biological knowledge accumulated over generations of observation. The sacred fig, Ficus religiosa, known as the Bodhi tree, is revered in both Hinduism and Buddhism. It was beneath a sacred fig that Siddhartha Gautama is said to have attained enlightenment. The species is genuinely remarkable, a strangler fig that begins its life as an epiphyte on a host tree, sending roots downward until they fuse and eventually engulf the host. It is one of the most ecologically important trees in tropical Asia, providing fruit to hundreds of species year-round even when other trees bear nothing. Celtic cultures held the oak in supreme reverence. The very word druid may derive from the Proto-Celtic word for oak. The oak was the tree of the sky gods, of Zeus, Jupiter, and the Norse Thor, associated with lightning perhaps because oak's deep root systems and tall stature make it among the most commonly lightning-struck of trees, a factual observation mythologized into divine favor. The yew, whose extraordinary longevity and habit of growing in churchyards made it a symbol of death and resurrection across northern European cultures, is now understood to contain compounds of genuine medicinal significance, the taxanes now central to cancer chemotherapy. The sacred reverence accorded to ancient trees in cultures worldwide often reflects an intuitive recognition of something that science has now confirmed, that exceptionally old trees are genuinely exceptional in measurable biological ways, not merely in their age. Ancient trees, whether Methuselah bristlecones, 1,000-year-old yews in Welsh churchyards, or the vast ancient beeches of Central European primeval forests, tend to support biodiversity far beyond what younger trees of the same species provide. Old-growth trees develop features that simply do not exist in young trees on any meaningful timescale: deep bark fissures that shelter specialized invertebrates, large cavities formed by centuries of decay that provide nesting sites for owls, bats, and hole-nesting birds unavailable elsewhere, the accumulation of decades of dead wood in the crown that hosts communities of rare wood decay fungi found nowhere else. The ancients who consecrated old trees, forbidding their cutting and designating them as sanctuary, were performing ecological conservation long before the concept existed as a formal discipline. The grove of sacred oaks at Dodona in ancient Greece, the temple groves preserved by Hindu communities across India, the sacred mountain forests maintained by communities in sub-Saharan Africa, many of these spaces have preserved old-growth biodiversity across millennia of surrounding landscape transformation. Stretching across northern Russia, Canada, Alaska, and Scandinavia in an unbroken belt that represents the largest biome on Earth, the boreal forest, or taiga, is a world of extraordinary austere beauty. It covers approximately 15% of Earth's land surface and contains an estimated one-third of the world's total forest carbon stock. Dominated by spruce, fir, pine, and larch, all conifers whose needle-like leaves and resinous wood are adapted to the brutal winters and short growing seasons of high latitudes, the boreal forest is far less species diverse than tropical forests but operates at scales of ecological function that dwarf its tropical counterparts in certain dimensions. The peatlands and permafrost soils beneath and adjacent to boreal forests have been accumulating carbon for thousands of years, locking it away in the form of partially decomposed organic matter that does not fully decay in the cold. These carbon stores are of global significance. The permafrost soils of boreal regions contain approximately twice as much carbon as is currently in Earth's atmosphere, and as climate warming accelerates, the thawing of these soils and their eventual decomposition releases carbon dioxide and methane in a self-amplifying feedback cycle that represents one of the most serious and least controllable aspects of contemporary climate change. At the opposite end of the thermal spectrum from the boreal taiga, the tropical rainforests of the Amazon basin, Congo, and Southeast Asia represent the apex of terrestrial biological complexity. Though they cover only about 6% of Earth's land surface, tropical forests harbor an estimated 50 to 80% of all terrestrial species, a concentration of life so dense that a single hectare of Amazonian forest may contain over 400 tree species, more than exist in the entirety of Europe north of the Alps. This diversity is not accidental or random but the product of a vast and intricate ecological network in which trees, animals, fungi, and microorganisms are bound together in webs of dependency and mutual shaping so elaborate that cutting a single species from the web can cascade through the system in unforeseen ways. The emergent trees of tropical rainforests, those individuals whose crowns rise above the main canopy, play a disproportionate ecological role, providing nesting sites for harpy eagles and habitat for hundreds of species of canopy epiphytes. Beneath the canopy, in the layered shade of the understory, grow palms, tree ferns, and broad-leafed angiosperms, each adapted with extraordinary precision to the spectral quality and intensity of the filtered light reaching them, creating what is effectively a vertical ecosystem of stunning complexity. At the margins of tropical and subtropical coastlines, in the intertidal zones where seawater and freshwater mingle in conditions of extreme salinity, waterlogged soil, and daily flooding, grows a community of trees so specialized in their adaptations that they constitute one of the most remarkable ecological systems on Earth. Mangroves, a term encompassing approximately 80 species from multiple independent evolutionary lineages, are trees that have evolved the ability to survive and thrive where virtually no other tree can exist. Their root systems are architectural marvels, aerial prop roots that arch above the waterline to access oxygen denied to roots buried in the anaerobic muck below, and pencil-like nematophores that protrude upward from the sediment-like living snorkels, allowing gas exchange with the atmosphere even as the rest of the root system is submerged. Mangroves excrete excess salt through specialized glands in their leaves, and their seeds, uniquely among trees, germinate while still attached to the parent, producing propagules that float and root themselves when they encounter suitable substrate. These ecosystems store carbon at rates per unit area far exceeding any other forest type, protect coastlines from storm surge and erosion, serve as nurseries for the majority of tropical fish species, and are disappearing at alarming rates from coastal development and aquaculture conversion. The annual spectacle of autumn color, the blazing reds, golds, and oranges that sweep across temperate forests in the northern hemisphere each fall, is among the most visually dramatic expressions of tree physiology. It is a phenomenon familiar to billions of people, and yet its chemical and ecological dimensions remain imperfectly understood. The yellows and oranges are the easiest to explain. They result from carotenoid pigments present in leaves throughout the growing season but masked by the abundant green of chlorophyll. As days shorten in autumn, trees begin the process of senescence, reabsorbing the nitrogen and other minerals locked in leaf tissue before shedding them. Chlorophyll, which requires magnesium and nitrogen and is energetically expensive to maintain, is broken down first, and as it retreats, the carotenoids beneath become visible. The reds are more intriguing. They result from anthocyanins, pigments actively produced in autumn leaves from sugars synthesized during the senescent phase. Why a tree would invest energy in making new pigments in leaves it is about to shed has been a genuine puzzle for plant physiologists, and several competing hypotheses have been proposed: that anthocyanins protect the leaf during resorption of nutrients from UV damage, or that they signal to aphids and other insects that the tree is nutritionally fortified and a poor host for their eggs. As autumn color fades and the last leaves fall, deciduous trees of temperate regions enter a state of dormancy so profound that their above-ground tissues must survive temperatures well below freezing, sometimes for months, without the metabolic activity needed to continuously repair cellular damage. How they accomplish this is a remarkable story of biochemical preparation. As days shorten and temperatures fall, deciduous trees receive the signal, primarily changes in day length perceived through phytochrome pigments in leaf tissue, to begin the transition to winter dormancy. They produce special proteins that prevent ice crystal formation within cells. They increase the concentration of sugars and antifreeze compounds in their cellular fluids, lowering the freezing point of their cell contents. They transform their cell membranes to remain flexible at low temperatures by altering their fatty acid composition. The carbohydrates stored in the roots and woody tissues provide energy for spring emergence without requiring any photosynthesis. Evergreen conifers, which do not shed their leaves in winter, instead harden their existing needles through similar biochemical changes, allowing them to photosynthesize on the rare warm days of winter while surviving the rest in a state of metabolic suppression. The buds that contain next year's leaves are sealed with protective scales and coated in resins, a preformed response to the coming season visible months before it arrives. The return of spring to a temperate forest is not a single event but a complex, carefully sequenced performance in which different species emerge in an order refined by millions of years of competition and coevolution. The timing of bud burst, the moment when a tree's dormant buds swell and open to reveal new leaves, is one of the most studied and ecologically significant events in forest phenology. Different species burst at characteristically different times, calibrated to a combination of accumulated warmth above a threshold temperature and the fulfillment of a cold requirement, a period of chilling temperatures needed to release dormancy and prepare the tree for spring growth. Ash and oak consistently break bud late among British woodland trees. Hazel and elder emerge early. The traditional English saying oak before ash, in for a splash, ash before oak, in for a soak, a weather predictor based on the relative timing of bud burst, reflects centuries of agrarian phenological observation. Modern research confirms that these timing differences have ecological consequences. Early emerging trees capture light before the canopy closes, while late emerging trees may avoid the worst of late spring frosts. Climate change is advancing bud burst dates across the northern hemisphere, disrupting the fine temporal calibration between tree emergence, insect hatching, and bird breeding that has evolved over millennia. Truly ancient, undisturbed forest, the kind that has never been felled, managed, or significantly altered by human activity, has become one of the rarest ecosystems on Earth. Less than 10% of the world's original forest cover remains in what ecologists classify as primary or old-growth condition, and much of what survives occupies terrain too steep, remote, or cold to have attracted commercial exploitation. The Białowieża forest, straddling the border between Poland and Belarus, represents perhaps the most studied remnant of lowland European primeval forest, a landscape of enormous linden, oak, elm, and hornbeam trees growing in conditions that have prevailed since the last ice age, their trunks clothed in mosses and lichens, their dead wood hosting communities of beetles and fungi found nowhere else in Europe. The Amazon and Congo basins preserve vastly larger areas of tropical primary forest, though these too are diminishing. In these places, the ecological processes visible in managed forests are amplified and enriched by time. Deadwood accumulates to depths and volumes that support entirely distinct communities of organisms. Soil fungal networks of extraordinary complexity and age develop. The trees themselves grow to dimensions rarely seen elsewhere, not just in height but in girth and structural complexity, their canopies hosting entire ecosystems of epiphytes, bromeliads, and mosses that would require centuries to re-establish after disturbance. The relationship between forests and rainfall is more profound and active than most people appreciate. Forests are not merely passive recipients of precipitation, they are, in a meaningful sense, generators of it. Through transpiration, the evaporation of water from leaf surfaces, large forest systems release enormous quantities of water vapor into the atmosphere. A single hectare of tropical forest may transpire over 50,000 liters of water on a warm day. This water vapor rises, cools, and condenses around particles released by the forest itself, the same volatile organic compounds that constitute phytoncides and other plant emissions serve as cloud condensation nuclei, seeding the formation of the clouds that then return rain to the forest below. In the Amazon basin, this process is so significant that researchers have identified atmospheric rivers of water vapor flowing inland from the Atlantic, augmented at each stage by forest transpiration in a cascade that transports moisture thousands of kilometers into the continent's interior. The trees of the Amazon do not merely depend on rainfall, they help create it. This discovery, formalized as the biotic pump hypothesis by Russian physicists Anastasia Makarieva and Viktor Gorshkov, has profound implications for deforestation policy. Removing forest not only eliminates the trees themselves but potentially disrupts the rainfall systems that sustain them and the agriculture that surrounds them. Soil, which we tend to regard as a fixed backdrop to life, is in fact one of its most dynamic and living products, and trees are among its primary creators. Rock becomes soil through physical, chemical, and biological weathering processes in which tree roots play a central role. Fine root tips, penetrating cracks in bedrock, physically pry rock apart as they grow. Root exudates, acids, enzymes, and chelating agents, chemically dissolve mineral surfaces, liberating elements otherwise locked in crystalline matrices. The fungi associated with tree roots extend this chemical weathering even further, with some mycorrhizal fungal species capable of dissolving silicate minerals that tree roots alone cannot breach. As tree leaves, bark, branches, and roots die and decompose, they add organic matter to the mineral substrate below, enriching it with nitrogen, phosphorus, and a complex of carbon compounds that feed the vast microbial community within the soil. The soil beneath a mature forest is a medium of extraordinary complexity, a living matrix of mineral particles, fungal hyphae, bacterial films, nematodes, earthworms, and organic matter in every stage of decomposition, bound together by fungal threads and polysaccharides secreted by bacteria. It took thousands of years of plant and tree life to build the rich agricultural soils of Europe and North America, soils that modern tillage agriculture is depleting at rates far exceeding the capacity for their natural renewal. Beyond chemical signaling through volatile compounds and mycorrhizal networks, trees are now known to transmit information through their tissues by means of electrical signals, a discovery that has drawn cautious comparisons to the nervous systems of animals. When a leaf is mechanically damaged, burned, or attacked by an insect, the cells at the injury site release ions that generate a wave of changed electrical potential that propagates outward through the plant's vascular system and parenchyma tissues, triggering defensive responses in distant, undamaged leaves. These action potential-like signals travel at speeds measured in centimeters per second, far slower than the millisecond speed signals of animal neurons, but capable of traversing an entire tree in minutes to hours. The mechanisms involve gated ion channels and the movement of calcium, hydrogen, and other ions across cell membranes in patterns that resemble, at least superficially, the electrochemical signaling of animal cells. Research groups in Germany and elsewhere have used sensitive electrode arrays inserted into living trees to record these electrical signals in real time, monitoring how a tree responds electrically to light changes, drought, temperature shifts, and wounding. The signals are genuine, measurable, and repeatable. Whether it is appropriate to draw deeper analogies to animal nervous systems remains a matter of considerable scientific and philosophical debate, but the electrical life of trees is no longer in doubt. The idea that trees might possess something analogous to intelligence, some form of information processing, adaptive response, and even memory that goes beyond the mechanical, has moved from the fringes of popular science toward the edges of mainstream botanical inquiry. The German forester Peter Wohlben's book, The Hidden Life of Trees, brought these ideas to a wide audience, and while some of his more anthropomorphic interpretations were contested by plant scientists, the biological phenomena he described were largely real. Trees clearly do process information. They integrate signals from light, temperature, gravity, touch, water availability, herbivore attack, and underground chemical signals and coordinate appropriate responses across their entire body. They modify their developmental trajectories in response to environmental history. Trees that have survived drought grow anatomically different wood in subsequent years, wood with smaller xylem vessels that provides greater resistance to embolism at the cost of reduced water transport efficiency. They appear to anticipate, producing defensive compounds before herbivores arrive based on airborne signals from neighbors. Whether these capacities constitute anything meaningfully analogous to cognition in an animal sense, or whether they are better described as sophisticated biochemical automation, is a question that depends as much on philosophy as on neuroscience, on what we ultimately decide the word intelligence should mean. While the coast redwood holds the record for tallest individual tree, its close relative, the giant sequoia, Sequoiadendron giganteum, is the world's largest tree by volume and by total mass, almost certainly by weight as well. The largest known giant sequoia is General Sherman, growing in Sequoia National Park in California's Sierra Nevada mountains. Its volume has been calculated at approximately 1,487 cubic meters, and its total mass is estimated at around 1,385 metric tons. Its base circumference exceeds 31 meters. Standing before General Sherman, a tree that was already 2,000 years old when Europeans first arrived in North America, induces a species of awe that has no precise analog in other natural experiences. Giant sequoias achieve these dimensions partly through their extraordinary longevity, typically living between 2,000 and 3,000 years, and partly through their habit of continuous, lifelong growth. Unlike many organisms that reach a maximum size and stop, sequoias continue to add wood to their trunks throughout their lives, producing rings that become narrower with age but never cease entirely. They also benefit from the fire-adapted ecology of the Sierra Nevada. Crown fires that kill competing species leave the fire-resistant sequoia standing, periodically clearing the competition and opening the mineral soil needed for sequoia seed germination. Among the most ecologically and culturally significant woody plants on Earth, bamboo occupies a paradoxical position in our botanical taxonomy. It is technically a grass, belonging to the family Poaceae, and yet many species grow to heights exceeding 30 meters and produce woody stems, culms, of remarkable structural strength. There are approximately 1,500 species of bamboo, distributed across tropical, subtropical, and temperate regions on every continent except Europe and Antarctica, and their ecological and economic importance is extraordinary. As a fast-growing material, bamboo can sequester carbon at rates per hectare exceeding those of most trees. Its rapid, continuous growth makes it a potentially valuable asset in carbon sequestration programs. Some tropical bamboo species are among the fastest-growing plants on Earth, with documented growth rates of nearly a meter per day under optimal conditions. Bamboo achieves this extraordinary growth rate not through cell division, as trees do, but through the rapid expansion of preformed cells within the culm, a strategy that allows the plant to allocate all its resources to elongation rather than the energetically costly process of producing new cells. The flowering biology of bamboo is one of nature's greatest mysteries. Many species grow for decades, some for over 100 years, flower once simultaneously across their entire global range, set seed, and die, in a synchronized event whose triggering mechanism remains incompletely understood. High in the canopies of tropical and subtropical forests, far above the forest floor, grows another forest, a community of plants that use the branches and trunks of trees as a substrate without parasitizing them, drawing their water and nutrients from rain, fog, and the thin layer of organic matter that accumulates on bark. These epiphytes, meaning literally upon plants, include orchids, bromeliads, ferns, mosses, liverworts, and lichens, and in the most biodiverse tropical forests they may account for more than half of all plant species in the community. A single large tropical tree can host hundreds of epiphyte species. Bromeliads trap rainwater in their central rosette, creating miniature aquatic ecosystems high above the forest floor, inhabited by tree frogs that breed there, microorganisms, and insect larvae found nowhere else. The roots of canopy epiphytes never reach the ground. Instead, they cling to bark with holdfast roots and absorb water through velamen, a spongy outer tissue that acts like a wick, drawing moisture from rain and fog with extraordinary efficiency. The accumulated organic matter and water held in epiphytic communities adds significant weight to forest canopy trees and represents a distinct ecological zone, with its own nutrient cycles, food webs, and microclimate, a world within a world, invisible from the ground and imperfectly explored even by science. Few sites in temperate and boreal forests are as visually striking as the incrustation of bark and branches with lichens, those gray-green, silver, rusty orange, or canary yellow growths that seem almost geological in their patient attachment to tree surfaces. A lichen is itself a composite organism, a partnership between a fungus and one or more photosynthetic partners, either green algae or cyanobacteria, and the relationship between lichens and their tree substrates adds yet another layer of biological complexity to the already layered ecology of the forest. Lichens are among the most pollution-sensitive organisms known, their sensitivity to sulfur dioxide and other air pollutants making them invaluable bioindicators of air quality. The luxuriance of lichen communities on tree trunks and branches in undisturbed ancient forests is itself a measure of ecosystem health. These slow-growing organisms may take decades to colonize new bark surfaces, and the dense cushions and pendulous streamers of lichen found on old-growth trees represent centuries of accumulation. They contribute to the tree's ecology in multiple ways, retaining moisture against the bark, cycling nutrients, providing nesting material and microhabitat for invertebrates. The gray-bearded streamers of usnea lichen that drape the branches of ancient oaks and conifers are among the most evocative features of the world's oldest forests, a visual shorthand for deep time. In managed forests, dead trees are typically removed as quickly as possible, for timber value, wildfire prevention, or simply from an aesthetic preference for tidiness. But in the ecology of natural forests, death is not an endpoint but a transformation, the beginning of a succession of biological processes that support extraordinary biodiversity and perform essential ecosystem functions. A single dead tree, from the moment it dies to its final incorporation into the soil, may pass through the territories of hundreds of species. Primary decomposers, wood decay fungi, are the first and most important agents of this transformation. These fungi, including the spectacular bracket fungi visible on standing dead wood, secrete enzymes that dissolve lignin and cellulose, converting what was structural wood into softer, more chemically accessible material. As the wood softens, bark beetles, longhorn beetles, and wood-boring wasps move in, and with them the woodpeckers, nuthatches, and small mammals that feed upon them. As the wood becomes yet more decayed and moist, it is colonized by specialist invertebrates, stag beetles, rhinoceros beetles, and hundreds of species of flies whose larvae develop only in very specific stages of wood decay. Eventually the remnant wood becomes humus, enriching the soil at a point that may be a century or more after the tree's initial death. Ancient forests preserve the full spectrum of this dead wood continuum. Most managed forests do not. Among the most ecologically valuable features of old trees are their cavities, hollows formed over centuries by a succession of processes beginning with the death of a branch. When a branch breaks or dies, the exposed heartwood is colonized by wood decay fungi that slowly hollow out the branch stub and eventually create a cavity within the trunk. These cavities, warmed by the sun, sheltered from rain, and insulated by the surrounding wood, provide nesting sites for a remarkable community of cavity-dependent species that cannot reproduce without access to suitable holes. In European forests, more than 60 species of birds are cavity nesters, including owls, woodpeckers, jackdaws, stock doves, and many small hole-nesting species like tits, nuthatches, and tree creepers, all of which require holes of specific sizes and depths. Mammals, bats, dormice, red squirrels, pine martens, similarly depend on tree cavities for resting and breeding. The supply of large, old trees with suitable cavities is among the most critical limiting factors for biodiversity in managed forests, and its scarcity is a primary driver of population declines in cavity-dependent species across the temperate world. A single ancient oak with multiple cavities, hollowing heartwood, and deep bark crevices may support hundreds of species, a biological treasure that takes at least 200 years to produce and is lost in minutes with a chainsaw. Determining the age of a living tree requires care, as the most straightforward method, counting annual rings in a cross-section, necessarily involves killing the individual. For valued or protected trees, dendrochronologists instead use an increment borer, a hollow, threaded instrument that extracts a thin cylindrical core of wood from the trunk to the center without causing significant damage. The core is removed, dried, fixed to a mounting board, and the rings counted under magnification. However, for truly ancient trees, this method has its own complications. Very old trees often develop rot in the heartwood that destroys the innermost rings, making direct counting impossible. In such cases, researchers must extrapolate, using the ring widths in the surviving outer core to estimate the radius that has been lost, and dividing by an average ring width. This is inherently approximate, and the ages cited for some of the world's oldest trees carry significant uncertainty. For clonal organisms like Pando, where no individual stem survives long enough to carry the full age record of the clone, geneticists instead use molecular clock methods and landscape modeling to estimate age. Carbon-14 dating of wood cells can also be employed in specific circumstances. The ages of ancient trees are therefore estimates with confidence intervals rather than precisely known quantities, a suitable epistemic humility when confronting the depth of biological time. For every ancient tree that has been formally identified, measured, and documented by science, there are almost certainly others that have not. The world's most remote forests, the deep interior of the Congo Basin, the unexplored river systems of the Amazon, the isolated valleys of New Guinea, the subalpine forests of Central Asia, have never been surveyed with the instruments and methods that could identify their oldest inhabitants. The bristlecone pine groves of the White Mountains were themselves discovered only in the mid-20th century, and Methuselah's identity was confirmed only in 1957. Before that, nobody knew that trees lived 5,000 years. There may be trees in places we have not yet looked with the appropriate attention that are older still, organisms carrying within their rings climate archives from before the end of the last ice age, before the founding of the first cities, before the beginning of recorded history. It is also worth acknowledging that our definition of individual tends to favor familiar morphologies. Clonal organisms like Pando challenge the concept almost to breaking point, and it is entirely possible that clonal systems in poorly surveyed landscapes, groves of trembling aspen in remote mountain regions, or networks of ancient oak root systems, harbor organisms of comparable or greater age that have never been identified as single entities. The forest, in this sense, still holds secrets we do not yet know how to ask for. The conifers, that ancient order of seed-bearing trees whose female cones give the group its name, represent one of the great evolutionary success stories of the terrestrial world. They appeared before the dinosaurs, diversified through the Mesozoic era when they dominated global forests, and though subsequently reduced in species diversity by the explosive radiation of the flowering plants, they have retained dominance over the most extreme terrestrial environments on Earth. No flowering tree can match the conifer's tolerance for dry, nutrient-poor soils. No angiosperm approaches the conifer's performance in the cold, short-growing seasons of boreal and subalpine environments. The secret lies partly in their evergreen habit. By retaining their needles for multiple years, conifers can begin photosynthesizing immediately when temperatures allow in spring, without waiting for new leaf development. Their needles are themselves marvels of engineering, deeply sunken stomata protected in grooves to reduce water loss, thick waxy cuticles that prevent desiccation, rounded cross-sections that shed snow without breaking under load, and resin canals that deter insects and seal wounds instantly. The world's oldest single trees, its tallest trees, and its most massive trees are all conifers, a quiet testament to the competitive superiority of a lineage that has been refining its design for over 300 million years. Among the most consequential roles played by the world's forests is their function as a global carbon reservoir, a biological store of carbon so large that its status fundamentally determines the composition of Earth's atmosphere and the pace of climate change. The world's forests are estimated to contain approximately 860 billion metric tons of carbon in their biomass, soil, and deadwood combined, more than double the amount currently in the atmosphere as carbon dioxide. Each year, through photosynthesis, forests absorb approximately 2.5 billion metric tons of carbon from the atmosphere, roughly a quarter of all anthropogenic carbon emissions, representing a carbon sink of incalculable value to the global climate system. This sink is, however, not permanent. Forests also release carbon through respiration, decomposition, fire, and logging. When a forest is cleared and burned, the carbon accumulated over decades or centuries is returned to the atmosphere within days. Tropical deforestation currently contributes approximately 10% of global carbon emissions annually. The balance between forest carbon uptake and release is shifting as climate change accelerates. Droughts, wildfires, and beetle outbreaks are converting previously carbon-absorbing forests into carbon sources in some regions, adding to atmospheric carbon dioxide in a feedback loop whose ultimate magnitude remains one of the most significant unknowns in climate science. Death comes to trees in as many forms as it comes to any organism, and often with a slowness that is itself a kind of life. Heart rot, the progressive digestion of heartwood by wood decay fungi entering through wounds or natural cavities, hollows trees from within over decades, eventually leaving only a thin shell of living sapwood standing against the wind. When the shell finally fails in a storm, the hollow reveals a century of interior decay, and within that decay, the records of the organisms that have lived in it. Lightning strikes kill many trees outright, but some large trees survive multiple strikes, their bark charred and their heartwood glassed by the current, yet their sapwood intact and functioning. Bark beetles, in epidemic numbers, can kill entire forests of pine and spruce within a few seasons, their tunnels girdling trees by destroying the cambium that connects root to crown. Fungal pathogens, Phytophthora, Armillaria, Hymenopsyphus, kill their hosts more slowly but with equal finality, and their spread through root contact and soil can convert healthy forests to standing ghostwood across vast areas. Drought kills trees by causing the water columns in their xylem to embolize, to snap under tension, filling with vapor and cutting off the flow of water to the crown. And eventually, inevitably, even the longest-lived trees succumb to the cumulative weight of cellular damage, hormonal dysregulation, and the attrition of biological time. A death without drama, the slow cessation of chemistry in wood. A forest is never static. Even when it appears to be in a stable, climax state, it is in reality engaged in a continuous slow process of species replacement, canopy gap creation, and competitive renewal that ecologists call succession. The concept describes the predictable sequence of community changes that occur over time as one assemblage of species prepares conditions for the next. Pioneer species colonizing bare ground, gradually modifying soil and microclimate until they are succeeded by longer-lived, more shade-tolerant species, which in turn succeed themselves over centuries in a process whose ultimate stable state, the climax community, is determined by regional climate, soil, and disturbance regime. The sequence varies by geography. In temperate Europe, birch and aspen colonize disturbed ground, followed by ash and oak, eventually giving way to beech in some regions. In boreal North America, lodgepole pine gives way to Engelmann spruce and subalpine fir. Each stage has its characteristic light environment, soil chemistry, and associated fauna. The intermediate stages are not mere stepping stones to the climax but valuable habitats in their own right, supporting species that depend on the structural heterogeneity of transitional forests. The ecologist's understanding of succession as a continuous, disturbance-driven process rather than a linear march toward a stable endpoint has deepened considerably since the concept was first formalized in the early 20th century. In the cities and towns where the majority of the world's human population now lives, trees face a uniquely hostile set of conditions that challenge their biology in ways forest trees never encounter. Urban soils are compacted by decades of foot traffic and construction, reducing pore space and impeding water infiltration, gas exchange, and root penetration. Road salts applied in winter leach into the soil and damage root tips. Hardscape surfaces prevent rain from infiltrating at all, leaving tree roots in a perpetual cycle of flood and drought. Air pollution, elevated temperatures from the urban heat island effect, and light pollution disrupting photoperiod responses all impose chronic physiological stress. Root systems confined by pavement, utilities, and foundations cannot explore the soil volume a tree of equivalent size would access in a natural setting, limiting water and nutrient acquisition. And yet, urban trees provide services to human communities that are both measurable and profound, reducing air temperatures through transpiration and shading, absorbing pollutants, attenuating stormwater runoff, reducing noise, increasing property values, and providing the documented psychological benefits of contact with living green. Cities worldwide are recognizing the value of their urban forests and investing in tree planting programs, but retaining mature street trees, which provide far greater ecosystem services than young replacements, requires engineering interventions, soil management, and institutional patience that do not come easily to municipal budgets. The relationship between trees and birds is among the most ecologically intricate and evolutionarily ancient partnerships in terrestrial ecosystems. Birds depend on trees for nesting sites, food, and shelter across virtually all forested habitats on Earth, and trees depend on birds for seed dispersal and insect population regulation in ways that have shaped the morphology and chemistry of both groups over millions of years. Frugivorous birds, those that feed on berries and fleshy fruits, are among the most important seed dispersers for a large proportion of tree species. The relationship between Calvaria major, the large-seeded Mauritian tree historically associated with the dodo, and the question of whether its seeds require passage through large animal guts for germination remains scientifically debated, but the broader dependency of many tree species on frugivore-mediated seed dispersal is undisputed. Insectivorous birds, meanwhile, provide a pest regulation service of quantifiable economic value. Studies have demonstrated that experimental exclusion of birds from forest plots results in significant increases in herbivore damage to foliage. The architecture of trees, branch angle, bark texture, cavity formation, is in part a product of selection pressures imposed by the foraging and nesting preferences of birds over evolutionary time. The singing of birds in a forest canopy is not merely beautiful, it is the acoustic surface of an ecosystem relationship of extraordinary depth. If birds are the most visible animal partners of trees, insects are unquestionably the most numerous and ecologically diverse. An estimated 10 million insect species inhabit terrestrial ecosystems, and the majority of them interact with trees in some capacity, as pollinators, herbivores, parasites, decomposers, or consumers of the organisms that parasitize trees. The relationship between oak trees and insects has been particularly well studied in Britain, where native oaks are estimated to support over 2,000 associated insect species, a number that dwarfs that of any other native tree. This extraordinary insect diversity reflects tens of millions of years of coevolutionary arms racing. As oaks evolved chemical defenses, insects evolved detoxification mechanisms. As insects evolved countermeasures, oaks evolved new compounds. The result is an arms race whose current equilibrium supports an insect fauna of remarkable specificity, with many species feeding only on oak and on no other host. Gall wasps represent one of the most intriguing chapters of this coevolution. These tiny insects inject chemical signals into oak tissue that redirect the tree's own developmental programs into the production of elaborate, architecturally precise galls, perfectly constructed chambers within which the gall wasp larvae develop, sheltered and provisioned by the hijacked chemistry of the oak itself. The oak produces hundreds of gall types, each created by a different species of gall wasp, each architecturally distinct. Along the banks of rivers and streams, where soil is periodically flooded and water tables are high, grow the riparian forests, communities of trees so intimately adapted to the flow of water that they have become integral components of the hydrological system itself. Willows, alders, cottonwoods, and river birches form the characteristic vegetation of stream margins in temperate regions, their roots binding the riverbank against erosion, their overhanging canopies shading the water and moderating the temperature fluctuations that stress cold-water fish. Alder, uniquely among temperate trees, has evolved a nitrogen-fixing symbiosis with the Actinobacterium frankia. Nodules on alder roots harbor these bacteria, which convert atmospheric nitrogen gas into forms the tree can use, enriching the poor alluvial soils of stream margins in a process analogous to the nitrogen fixation performed by legume-rhizobium partnerships in agricultural soils. The leaf litter and woody debris that falls from riparian trees is a primary energy input to river food webs. The invertebrates that shred and consume this leaf litter fuel the aquatic food chain from insects to fish. When riparian trees are removed, stream temperatures rise, bank erosion accelerates, fish populations decline, and the flood-attenuating capacity of the riverine landscape is diminished. The re-establishment of riparian woodland along degraded watercourses is one of the most cost-effective ecological restoration interventions available to land managers. Few subjects have occupied human artistic imagination as persistently as trees. From the earliest known cave paintings, which sometimes depict plants, to the landscape art of the 19th century, to the modernist abstraction of Georgia O'Keeffe and the ecological art of Andy Goldsworthy, trees have served as symbols, subjects, and structural metaphors for every artistic tradition that emerged in a forested landscape. John Constable's monumental canvases of English oak woodland captured something about the specific quality of light beneath a broad canopy that resonated across Europe in the early 19th century. Vincent van Gogh's Starry Night olive trees, writhing, expressive, almost animate in their forms, project onto the tree a psychological turbulence that is among the most recognizable expressions of the meeting between inner life and observed nature in Western art. In literature, trees appear as structural metaphors in their most literal sense: genealogical family trees, the tree of knowledge, the tree of life, but also as presences charged with emotional meaning, the tree of childhood, the forest as the space of wildness and freedom beyond the civilized clearing, the ancient yew in the graveyard as a portal between living and dead. Tolkien's Ents, ancient walking trees with memories of the deep forest past, represent perhaps the most elaborate literary elaboration of the intuition, now partially validated by science, that trees are somehow more than passive objects in the landscape of life. The application of tree-ring science to archaeology has produced some of the most precise and dramatic dating achievements in the history of the discipline. Because wood preserves well under certain conditions, in the dry environments of Egyptian tombs, in the waterlogged anaerobic muds beneath ancient lake villages, in the cool interiors of historic buildings, dendrochronologists can extract cores from ancient timbers and match their ring patterns to the master chronologies built from living and subfossil trees to determine not just the decade but often the precise year in which a tree was felled. The precision is extraordinary. A barn or cathedral beam can be dated to within a year or two, and in some cases the dating reveals not just the age of the timber but the season of its felling, since the outermost ring of freshly cut wood is often incomplete. This technique has been applied to date Viking longships, medieval bridges, Egyptian sarcophagi, Bronze Age lake dwellings in the Swiss Alps, and the timbers of historic ships recovered from the seabed. It has confirmed disputed attributions of paintings on wooden panels, demonstrated that some purported ancient artifacts are forgeries, and precisely dated volcanic eruptions visible in the ring record as narrow frost rings. The intersection of dendrochronology with radiocarbon dating has produced the most accurate calibration of the radiocarbon timescale available, a gift from trees to the archaeologists who study the civilizations that grew in their shade. To attempt to catalog the role of trees in human civilization is to attempt to catalog civilization itself. The debt is so deep and so pervasive as to be nearly invisible, the way the debt to air is invisible to a creature that has never been without it. Wood was the primary building material of every human civilization until the industrial age. Houses, ships, wheels, tools, Furniture, bridges, fortifications, fuel, all wood. The great naval empires of history, Phoenician, Greek, Roman, Spanish, British, were built on access to specific timber resources, and their expansion was often fundamentally driven by the need to secure new forests as original ones were depleted. The deforestation of the Mediterranean basin by successive civilizations, from the Levant cedar forests logged for the palaces of Mesopotamia to the oaks of Greece and Italy felled for ships and charcoal, represents one of the earliest and most consequential human-driven environmental transformations. Charcoal smelting, the conversion of or to metal using wood as the reducing agent, drove forest clearance across Europe and Asia for thousands of years before coal became available. Writing itself, in both its papyrus-based Egyptian form and the parchment traditions of Europe, depended on plant material. The codex replaced earlier wooden writing tablets. The history of every material dimension of civilization runs through wood. Among the most consequential industrial uses of trees is the production of paper, whose invention in China around the 2nd century BCE and whose eventual global diffusion transformed human knowledge systems by making written information cheap enough to circulate widely. Before paper, text was inscribed on clay tablets, papyrus reed sheets, animal skin parchment, or silk, all materials that were scarce, expensive, or fragile. Paper made from plant fiber, initially hemp and mulberry bark and later predominantly wood pulp, could be produced in enormous quantities at low cost, and the combination of paper with printing technology produced the information revolution of the 15th century, whose effects on the distribution of power, knowledge, and eventually the structure of societies were as radical as the internet has been to ours. Today, global paper production consumes approximately 4 billion trees annually, and though digital technology has reduced demand for printing paper in some markets, packaging paper demand continues to grow with the expansion of global e-commerce. The ecological footprint of paper production is significant. Beyond the trees themselves, pulp mills are major water users and have historically been significant sources of water pollution. Certified sustainable forestry for paper production, recycled fiber use, and digital substitution are reducing but have not eliminated the forest pressure that paper demand represents. As the scale of global deforestation and its consequences for climate, biodiversity, and water security has become increasingly clear, large-scale tree planting has emerged as one of the most widely discussed and contested strategies in environmental policy. Reforestation, the replanting of trees on land that was previously forested, and afforestation, the planting of trees on land that has not historically been forested, are both being pursued at enormous scales globally. China's 3 North Shelter Forest Program, initiated in 1978 and extending to 2050, aims to plant a belt of trees across northern China to arrest the expansion of the Gobi Desert, the so-called Great Green Wall, now encompassing tens of millions of hectares. Ethiopia reported planting over 300 million trees in a single day in 2019 as part of a national restoration program. Yet the scientific community has raised significant concerns about the design and implementation of many tree planting initiatives. Plantations of fast-growing monocultures, eucalyptus and acacia are common choices in tropical programs, can deliver very different ecological outcomes than diverse, naturally regenerating forests, often providing minimal biodiversity benefit and even, in some cases, reducing local water availability through the high transpiration of productive monocultures. The most ecologically effective and durable forest restoration, researchers increasingly argue, is the protection of naturally regenerating forest, which rebuilds ecological complexity far more effectively than any planting program. For most of human history, the distinction between forest and farmland was far less absolute than modern landscape management implies. Traditional agricultural systems around the world have long incorporated trees into food production landscapes, creating diverse, multi-layered systems that simultaneously produce food, timber, and fuel while maintaining soil fertility, moderating microclimate, and supporting biodiversity at levels far above those of treeless monoculture. These systems, now collectively known as agroforestry, are receiving renewed scientific and policy attention as evidence accumulates of their multiple benefits relative to conventional agriculture. Tropical agroforestry, the growing of coffee, cacao, and other shade crops beneath a canopy of diverse shade trees, maintains bird diversity, soil carbon, and hydrological function far more effectively than sun-grown monoculture plantations. Silvopastoral systems combine grazing animals with trees on the same land, the trees providing shade that reduces heat stress in livestock, leaf litter that fertilizes the soil, and structural diversity that maintains soil carbon and water holding capacity. In temperate Europe, traditional orchard meadows, pastures dotted with widely spaced fruit trees, support exceptional biodiversity including rare lichens, beetles, and bat species that depend on old fruit tree cavities. The recognition that trees in farmed landscapes are not obstacles to productivity but contributors to it represents a significant shift in agricultural thinking with profound implications for how we manage the landscapes that feed the world. Certain tree species function as what ecologists call keystone species, organisms whose ecological impact is disproportionately large relative to their abundance, and whose removal causes cascading changes that transform entire ecosystems. In temperate forests, few species exemplify this better than the beaver, but the trees the beaver depends on, willows and aspens, are themselves keystone organisms whose presence determines the character of riparian and wetland ecosystems. In tropical forests, large fruiting trees that produce fruit year-round during periods when other food sources are scarce, what ecologists call keystone resources, are disproportionately important to the maintenance of frugivore communities. When these trees are selectively logged, their loss propagates through the food web, reducing frugivore populations and consequently reducing seed dispersal rates for the many tree species that depend on those frugivores, ultimately simplifying the tree community. More broadly, the physical structure of trees, the architecture of their canopies, root systems, and deadwood, creates and modifies habitat for hundreds of other species in ways that constitute a form of ecological engineering. The bole of a large old tree is habitat, its crown is habitat, its deadwood is habitat, its root system creates habitat, its leaf litter creates habitat. The removal of large old trees from a forest is therefore not simply the loss of one tree but the collapse of a microecosystem that may have taken centuries to develop. Trees fail, they break, uproot, and collapse. And the physics of this failure reveals much about the engineering principles that govern their growth. A standing tree must resist not only the vertical force of its own weight but the horizontal forces imposed by wind, which can generate bending moments at the base that are orders of magnitude larger than the tree's own mass would suggest. The tapering form of tree trunks, which are thicker at the base and narrower toward the crown, is precisely the form required to maintain approximately constant stress along the length of a column under the bending loads imposed by wind, a result derived independently by mechanical engineers studying optimal beam design and found to be closely matched by measurements of real trees. Trees fail in two distinct modes: crown breakage, in which the upper trunk or major branches snap, typically in wood weakened by decay, and uprooting, in which the root plate tears from the soil, the tree pivoting about a fulcrum at the windward edge of the root plate. Shallow-rooted trees in waterlogged soils are most susceptible to uprooting. Trees with decayed heartwood are most susceptible to stem breakage. The aftermath of a large tree windfall is itself ecologically significant. The pit and mound microtopography created by the upturned root plate provides distinctive habitat for many soil-dwelling invertebrates and fungi, and the fallen crown, in decomposing over decades, creates a nurse log substrate for the germination and growth of the next generation. The role of mycorrhizal networks in supporting forest regeneration, particularly the establishment of seedlings beneath closed canopies where light is too limited for independent photosynthetic survival, represents one of the most important and least appreciated aspects of these subterranean partnerships. Seedlings establishing beneath the dense shade of a mature forest canopy are in a physiological bind. They require light to photosynthesize and produce the sugars needed for growth, but so little light reaches the forest floor that their own photosynthesis cannot sustain their metabolic needs. Research has demonstrated that many such seedlings survive this critical early life stage by receiving carbon subsidies through mycorrhizal connections to larger, canopy-layer trees, essentially being fed by their neighbors through the fungal network. This carbon transfer can make the difference between seedling survival and death in the critical months following germination. The fungal network therefore acts as a kind of forest infrastructure for regeneration, an underground system that helps buffer the extreme microenvironmental heterogeneity of the forest floor and maintains a supply of juvenile trees capable of responding to canopy gaps when they open. Disturbances that damage mycorrhizal networks, heavy mechanical logging, deep plowing, fungicide application, severe drought, can impair forest regeneration for years or decades by eliminating the fungal partnerships that new trees need to establish. Natural regeneration in disturbed forests depends critically on the survival of even small remnant patches that preserve functional mycorrhizal networks. Among the most ecologically and economically significant tree-form plants in tropical and subtropical regions are the palms, technically monocots rather than true trees, but producing woody, upright stems that can reach 40 meters or more and live for decades. With approximately 2,700 species distributed across tropical and subtropical regions, palms are among the most economically important plant families on Earth. The oil palm alone currently provides 40% of the world's vegetable oil. Coconut palms provide food, fiber, and timber to millions of people across the Pacific and Indian Ocean regions. Date palms have sustained human populations in arid North Africa and the Middle East for 6,000 years. Biologically, palms differ from true trees in fundamental ways. They grow from a single apical meristem rather than through cambial activity, meaning they cannot repair a damaged trunk by growing over it. A palm whose crown is destroyed is dead. They also lack true secondary thickening, instead producing a trunk of essentially constant diameter by rapidly adding cells to the base of the stem during the early years of growth. The fan-like or feather-like fronds of palms are among the largest individual leaves in the plant kingdom, with some species of raffia palm producing leaves over 25 meters long, the longest of any plant. Of all the thousands of tree species in tropical forests, perhaps none plays as pivotal an ecological role as the figs, those plants of the genus Ficus, comprising approximately 850 species of trees, shrubs, and climbing plants distributed across tropical and subtropical regions worldwide. Figs are extraordinary for many reasons, but their ecological centrality stems above all from their phenology. Individual fig trees of many species fruit asynchronously from all other figs, so that somewhere in any large tropical forest, a fig tree is always in fruit. This means that figs provide food during the lean seasons that would otherwise decimate populations of frugivores, the birds, bats, monkeys, and fruit pigeons that depend on fruit. Figs are therefore among the most critical keystone resources in tropical forest ecosystems. Their reproductive biology is itself one of nature's most extraordinary stories. Each fig species has a specific obligate mutualist, a species of tiny wasp from the family Agaonidae, whose female enters the fig through a narrow pore, pollinates the flowers inside, lays her eggs, and dies within the fruit. The wasp larvae develop, mate, and the wingless males die inside the fig while the fertilized females emerge, collect pollen, and fly to a new figure. Neither fig nor wasp can reproduce without the other, a coevolutionary lock of mutual dependency refined over 90 million years. The effects of contemporary climate change on the world's forests are already measurable, significant, and in many regions deeply alarming. The global mean temperature has risen approximately 1.2 degrees Celsius since pre-industrial times, but the warming signal is not uniform. High-latitude boreal and mountain forests have experienced warming several times the global average, driving range shifts in tree species, advancing phenological events, and disrupting the disturbance regimes that have structured these ecosystems for millennia. Bark beetles in the coniferous forests of western North America provide perhaps the most dramatic current example. Warmer winters have reduced mortality of overwintering beetle larvae, allowing population explosions of mountain pine beetle, spruce beetle, and others that have killed tens of millions of hectares of forest, areas visible from space as swaths of red, dead canopy. Longer droughts, combined with warming-induced stress, have increased the susceptibility of forests to both beetle attack and wildfire. Forest fires in western North America, Australia, Siberia, and the Amazon have grown in frequency, extent, and severity over the past 2 decades, in patterns that climate projections suggest will intensify. Range shifts are documented across multiple tree species, with the leading edge of species distributions moving northward and to higher elevations in response to warming, while trailing edge die-offs are occurring in the southern portions of ranges where summer heat and drought now periodically exceed tolerance thresholds. The value of tree ring records as archives of past climate extends well beyond the question of whether the current warming is unusual in historical context, though they speak eloquently to that question as well. The longest continuous tree ring chronologies, built by combining data from living trees with preserved subfossil wood recovered from bogs, lake sediments, and ancient buildings, extend back over 10,000 years in some regions of Europe and North America. These records have been used to reconstruct summer temperatures, precipitation patterns, drought severity, and even sea surface temperatures in regions where ocean-atmosphere teleconnections influence terrestrial climate. The medieval climate anomaly, visible in tree ring records as a sustained period of above-average growth in many northern regions between approximately 950 and 1250 CE, was both real and spatially complex, with different regions showing different timing and magnitude of warmth. The Little Ice Age, spanning roughly 1300 to 1850 CE, is equally visible in ring records and was also spatially heterogeneous. Both events, when placed in the context of the full 10,000-year tree ring record, appear as modest fluctuations compared to the sustained warming of the 20th and 21st centuries, a comparison that has proven consequential in climate science, establishing that the current warming trajectory is unprecedented in at least the Holocene era. The trees, patient and meticulous as ever, have recorded the departure from normal in their narrowing rings. Among the most carbon-dense ecosystems on Earth, mangrove forests occupy a special place in the emerging science of blue carbon, the carbon stored in coastal and marine ecosystems. Per unit area, mangrove soils store more carbon than virtually any other ecosystem type, accumulating organic matter in their waterlogged, anaerobic soils at rates that reflect both the high productivity of the trees themselves and the very slow decomposition rates in oxygen-depleted conditions. The estimated total carbon stored in mangrove soils worldwide is somewhere between 4 billion and 20 billion metric tons, a range whose uncertainty reflects how poorly these ecosystems have been mapped and measured, but even the lower estimate is ecologically significant. When mangroves are cleared, for shrimp aquaculture ponds, resort development, or timber, this carbon is rapidly oxidized as the soils drain and aerate, releasing centuries of stored organic carbon as carbon dioxide in a process that can take years to decades and cannot be easily reversed. The conversion of mangroves to aquaculture has been one of the most destructive ecological transformations of recent decades. Approximately a third of the world's mangrove forests have been destroyed since 1950, releasing not only their above-ground biomass carbon but the enormous stocks of ancient soil carbon beneath. Mangrove restoration is consequently among the highest carbon benefit ecological investments available in tropical regions, with the added dividends of coastal protection, fisheries nursery habitat, and biodiversity conservation. The technical challenges of successfully establishing trees in degraded or novel environments are considerably greater than they might appear to an optimistic policymaker or celebrity tree planting advocate. A seedling planted in degraded soil without appropriate mycorrhizal fungi cannot access the nutrients its survival depends on. A species planted outside its natural climatic range will struggle with mismatched phenology and pest regimes. A planting of a single species across a large area creates a monoculture as vulnerable to catastrophic failure from disease or pest outbreaks as any agricultural crop. The ecology of tree planting is a technically demanding science, and the history of large-scale afforestation is littered with failures. Vast plantations of Sitka spruce in upland Britain that excluded biodiversity and acidified streams, eucalyptus monocultures in Africa that dried up water sources, invasive tree species introduced for shade or timber that have become ecological problems. The growing consensus among restoration ecologists is that the most durable and ecologically effective approach combines the protection of naturally regenerating vegetation, allowing trees to establish from local seed sources already adapted to local conditions, with appropriate mycorrhizal partners and insect communities, with targeted planting of specific missing species where natural regeneration is insufficient. This approach, sometimes called assisted natural regeneration, consistently outperforms intensive planting in long-term ecological outcomes while being considerably less expensive. The genetic diversity of tree populations is of profound ecological and evolutionary significance, representing the raw material from which adaptation to changing conditions can be assembled over generations. Unlike annual plants, which can complete many generations in a human lifetime, trees cycle through generations on timescales of decades to centuries, meaning that their evolutionary response to rapidly changing environmental conditions is necessarily slow relative to the pace of climate change. This makes the maintenance of genetic diversity within tree populations especially critical. A genetically uniform population, whether from natural founder effects or human reforestation using seeds from a single source, may lack the heritable variation needed to respond adaptively to novel temperature extremes, disease pressures, or drought regimes. Research into local adaptation in trees has revealed that populations from different parts of a species range often differ genetically in traits relevant to survival: bud burst timing, drought tolerance, frost hardiness, disease resistance, calibrated to the specific conditions of their home environment. These locally adapted genotypes may perform poorly when transplanted to different climates, a phenomenon called maladaptation. Forest geneticists are consequently developing the concept of assisted gene flow, deliberately moving seeds or pollen from warmer, drier parts of a species range to cooler, wetter populations that will eventually experience the conditions those genotypes are already adapted to. The mycorrhizal networks that underpin forest health and regeneration are themselves under threat from the same combination of environmental changes that stress the trees they serve. Nitrogen deposition from agricultural and industrial sources is among the most pervasive threats to mycorrhizal fungi in temperate forests. Elevated nitrogen availability shifts the economics of the tree-fungal relationship, reducing the tree's dependence on fungal nutrient supply and consequently reducing the carbon subsidies it allocates to its fungal partners. Studies in polluted European forests have documented significant reductions in mycorrhizal fungal diversity and abundance associated with nitrogen deposition, with potential long-term consequences for forest nutrition and resilience. Drought, increasingly prevalent in many forest regions, can disrupt mycorrhizal networks by killing or fragmenting fungal mycelium, reducing the extent of the underground connections. Pesticide application in forests and adjacent agricultural land can directly kill non-target fungal species, and the broad-spectrum fungicides used in some disease management programs are particularly damaging. Forest fragmentation, by reducing the pool of tree species and the area available for fungal network development, may reduce the diversity and connectivity of mycorrhizal networks in the same way that habitat fragmentation reduces animal population viability. The subterranean infrastructure of the world's forests is as threatened as its canopy, and rather less visible to the people who make decisions about land management. The fallen logs and branches of riparian trees, what hydrologists call large woody debris, play a role in river ecosystems so fundamental that their removal over the past 2 centuries has silently transformed the character of rivers across the industrialized world. Large wood in rivers creates hydraulic complexity. It deflects current, creates pools, traps sediment and organic matter, and generates the structural diversity of stream habitat that supports the most diverse and abundant communities of aquatic invertebrates and fish. Salmon and trout both depend on the pools formed by large wood for resting, feeding, and spawning habitat. The invertebrate communities that develop in the leaf packs trapped against submerged logs are among the most ecologically important in stream systems, processing leaf litter and cycling nutrients through the food web. When European and North American rivers were cleared of snags, woody debris that impeded navigation and was believed, incorrectly, to cause flooding, the unintended ecological consequences were profound. Rivers straightened, pools disappeared, gravel beds scoured clean, and the biological productivity that had sustained the world's greatest salmon and trout fisheries collapsed over decades. The reintroduction of large woody debris to rivers, sometimes by dragging in logs, more effectively by restoring the riparian woodland that continuously supplies it, is now recognized as one of the most effective river restoration interventions available, restoring ecological function that was lost to well-intentioned engineering. The The fossil record of trees extends far enough into the past to reveal not just the origins of recognizable tree forms but the existence of entire forest ecosystems so different from anything alive today that they constitute a kind of alien biology. The Devonian fossil forest site at Gilboa, New York, preserves the stumps of some of the oldest known trees, Eospermatopterus, plants that bore no resemblance to modern trees in their internal structure, reproducing through spores and possessing trunks more like giant club mosses than any familiar woody plant. In the Carboniferous period that followed, the world's forests were dominated by scale trees, Lepidodendron and Sigillaria, giant lycopods whose trunks grew to heights of 30 meters and whose surfaces were marked by the distinctive diamond-shaped leaf scar patterns visible in Carboniferous coal measures worldwide. These plants reproduced through spores rather than seeds, and unlike modern trees whose bark contains substantial lignin, their trunks were so low in lignin that the wood decay fungi capable of decomposing lignin had not yet evolved, meaning that fallen Carboniferous trees did not decompose, instead accumulating in vast peat swamps whose compression over millions of years produced the coal deposits that powered the Industrial Revolution. The Carboniferous was, in this sense, an anomaly created by a biochemical arms race between tree evolution and microbial decomposition, a race the fungi eventually won, ending the coal-forming era. Among the most dramatic and biologically fascinating growth strategies in the plant kingdom is the habit of the strangler figs, a loosely defined ecological group of ficus species that begin their lives as epiphytes in the canopy of a host tree and end by surrounding, engulfing, and in many cases killing that host. The process begins when a bird or fruit bat deposits a fig seed in the canopy of a large tree, in a crevice or epiphytic mat where organic matter has accumulated. The seed germinates and the young fig grows as an epiphyte for years, sending aerial roots downward along the host trunk. When these roots reach the soil, they anchor the fig to the ground, and growth accelerates. More roots are sent down, they thicken, fuse, and anastomose join, to form a latticed shell that progressively encloses the host trunk. Meanwhile, the fig's canopy expands above the hosts, shading it out. Eventually, the host dies and decays, leaving a hollow cylinder of fused fig roots that stands independently. A strangler become a freestanding tree, its victory complete. The ecological importance of strangler figs extends beyond this spectacular narrative. The hollow trunks they produce when the host dies become among the most valuable cavity habitats in tropical forests, sheltering bats, small cats, pythons, and a remarkable diversity of smaller fauna within the latticed wooden architecture of their violent success. Throughout human history and across virtually every culture, trees have been among the primary sources of the medicines, remedies, and psychoactive substances that humans have administered to one another in sickness and in ceremony. The long list of tree-derived pharmaceuticals currently in clinical use represents only the most recently validated fraction of a medicinal tradition extending back beyond any written record. Aspirin, still the world's most widely used drug, derives from salicylic acid, first isolated from willow bark, which has been used in traditional medicine for at least 4,000 years. Quinine, extracted from the bark of the cinchona tree of South America, was the first effective treatment for malaria and remained the primary anti-malarial drug until the 20th century. Morphine and codeine are derived from the opium poppy, a herbaceous plant, but many other opioid pathway compounds come from tree species. Taxol, perhaps the most significant cancer chemotherapy agent of the late 20th century, was isolated from the bark of the Pacific yew, a slow-growing tree once regarded as worthless by the timber industry. Artemisinin, extracted from Artemisia annua, now supplies the most effective anti-malarial drugs in use. The ethnobotanical record of traditional forest medicine systems, Ayurveda, Chinese medicine, Amazonian plant knowledge, remains a vast, incompletely explored archive of biologically active compounds embedded in the tissues of trees that evolved them over millions of years. The forest that presents itself to the daytime visitor, a world of birdsong, dappled light, and visible movement, is only half the story. As dusk falls and the light fades from the canopy, a transformation occurs in the forest that is no less complex or ecologically significant than what happens by day, and in some respects more so. The trees themselves alter their metabolism as darkness arrives. Stomata close across millions of leaf surfaces, reducing water loss and temporarily halting gas exchange. The electrical activity detectable in the vascular system of trees shows measurable diurnal variation, with some researchers describing patterns that resemble, very loosely, the sleep rhythms of animals. Bats emerge at dusk to begin the pollination of night-blooming flowers in tropical forests. Many of the most important tropical trees, including multiple species of durian and baobab, are bat-pollinated, and to consume the insects that constitute one of the forest's most significant herbivore guilds. Moths, many of which fly and pollinate only at night, are active throughout the dark hours, performing pollination services for a distinct subset of forest plants, particularly orchids. The forest floor becomes the territory of badgers, hedgehogs, wood mice, and a host of nocturnal invertebrates that remain hidden through the day. The fungi that decompose dead wood grow and sporulate most actively at night in humid conditions. The forest does not sleep, it merely changes shift. For indigenous communities living within forested regions around the world, the accumulated ecological knowledge of trees, their uses, behaviors, seasonal cycles, relationships with animals, and management requirements, represents a vast body of understanding developed over thousands of years of intimate observation and refined through generations of practical engagement. This indigenous ecological knowledge is, in many cases, scientifically validated and sometimes extends further in certain directions than formal scientific study. Indigenous communities in the Amazon have recognized the network-like connectivity of forest plants for centuries, encoded in cosmological frameworks that foreground the relationality of forest organisms rather than treating them as independent individuals. Aboriginal Australian peoples possess detailed knowledge of fire ecology and have managed forests and woodlands through controlled burning for at least 50,000 years, a practice whose ecological value, long dismissed by colonial forestry, is now recognized as maintaining the fire-adapted biodiversity of Australian landscapes. Forest communities in Central Africa and Southeast Asia possess pharmacological knowledge of tree species that regularly yields novel bioactive compounds when tested by pharmaceutical researchers. The current destruction of forests worldwide is simultaneously the destruction of the land-based cultures whose ecological knowledge depends on those forests and who hold knowledge about those forests not yet contained in any scientific database. The loss of indigenous forest knowledge systems is a form of intellectual extinction concurrent with the biological extinctions of the forests themselves. Among the most sensitive biological indicators of environmental change available to ecologists are the lichen communities of tree bark and branches, organisms that accumulate in old-growth forests over centuries and disappear rapidly in response to air pollution, habitat disturbance, and microclimate change. The oceanic epiphytic lichens that drape the branches of ancient oak and beech in the hyperoceanic temperate rainforests of western Scotland, Ireland, and Brittany represent some of Europe's rarest and most threatened ecological communities, dependent not only on clean air and high humidity but on the very specific microclimate created by large, old trees with deeply furrowed bark. These lichen communities, known as Atlantic or Celtic rainforest lichens, can be used as precise indicators of habitat continuity. Species like Lobaria pulmonaria and Stictolimata are found almost exclusively in sites with centuries of unbroken canopy cover, unable to recolonize once lost even if suitable trees regrow. The rate of lichen recolonization on restored woodland sites is so slow, decades to centuries for the most sensitive species, that lichen communities serve as something approaching a clock for ecological continuity, recording the depth of a site's history in its own structural complexity. The painstaking cataloging of lichen diversity on ancient trees has consequently become a valuable tool for identifying forest sites of exceptional conservation value that would not be identified by conventional surveys of vascular plants or vertebrate animals. The oxygen in every breath you take was put there, in its current abundance, by photosynthetic organisms. And trees have been among the most significant contributors to maintaining that abundance for the past 350 million years. The emergence of land plants and their explosive diversification during the Carboniferous period dramatically elevated atmospheric oxygen concentrations, reaching levels perhaps as high as 35% compared to the current 21%, conditions that made enormous insects possible and fed the coal-forming fires that left their record in ancient charcoal deposits. Modern forests contribute approximately 50% of all terrestrial photosynthesis, producing oxygen as a byproduct of carbon fixation. A single mature beech tree produces enough oxygen annually for approximately 10 people. The Amazon basin alone produces approximately 20% of the oxygen generated by terrestrial ecosystems. However, a complete accounting of the forest oxygen budget must include respiration. Trees, like all aerobic organisms, consume oxygen in breaking down sugars for energy, and in a forest in steady state, one that is neither gaining nor losing carbon, photosynthetic oxygen production is precisely balanced by respiratory oxygen consumption. Forests contribute net oxygen to the atmosphere only when they are accumulating carbon, when they are growing. This is another reason why the net carbon sink maintained by the world's forests is significant. It represents not merely stored carbon but a genuine net contribution to atmospheric oxygen. At the upper limit of elevation where trees can survive, a boundary known as the treeline, trees encounter conditions of cold, wind, snow loading, and ultraviolet radiation that require the most extreme physiological and morphological adaptations in the forest repertoire. Treeline trees exist in a marginal zone where the growing season may be measured in weeks rather than months, where night frosts occur in every month of the year, and where wind chill, ice crystal abrasion, and desiccating winter winds create conditions as physiologically challenging as any hot desert. The characteristic form of treeline trees, the krummholz, a German word meaning crooked wood, is one of the most evocative and instructive shapes in nature. These trees, many of them centuries old, grow prostrate and layered against the ground, their windward branches killed by ice abrasion and their leeward branches growing close to the warm soil surface, forming dense mats that may be only knee-height but many meters across. Ring measurements from krummholz specimens often reveal growth of less than a millimeter per year, rings so fine they can only be counted under a powerful microscope. At the other extreme of latitude, in the subarctic zones beyond the boreal tree line, black spruce clings to the permafrost in dwarf, spire-like forms whose entire exposed surface above the snow line is killed each winter, surviving only through the insulation of accumulated snow. A forest is never truly silent. Even on the stillest day, with no wind to stir the leaves and no birds calling in the canopy, the forest produces sounds so subtle and so closely interwoven with the natural acoustic background that they pass below the threshold of conscious attention, yet are measurably present in the sonic environment that shapes the behavior and physiology of every organism within it. The creak of wood under thermal stress, the expansion and contraction of trunks as temperature changes, is one of the most characteristic sounds of old forests in cold weather. Research on the physics of cavitation in xylem, the snapping of water columns under tension during drought, has documented that embolisms produce ultrasonic clicking sounds in stressed trees, inaudible to human ears but detectable with sensitive microphones, and potentially detectable by wood-boring insects that use these sounds to locate drought-weakened trees. The low-frequency sounds produced by wind moving through a forest canopy have been analyzed as a complex acoustic environment in which bird songs have evolved specific frequency ranges and temporal patterns to be maximally detectable above the ambient noise, leading to documented divergence in bird song structure between forest and open habitat populations. Even the growth of roots through soil and the activity of mycorrhizal fungi have been detected as faint acoustic signals by instruments sensitive enough to register them. The forest, in this reading, is continuously broadcasting. Not all plants that adopt the form of trees are entirely independent in their nutrition. A remarkable and often overlooked group of tree-form plants are the hemiparasites, species that are photosynthetically active and can survive independently to varying degrees, but that also tap into the root systems or xylem of neighboring trees to supplement their water and mineral supply. The mistletoes are the most familiar examples. Those iconic evergreen clusters that perch in the branches of oaks, apples, and limes across temperate regions are not epiphytes but hemiparasites. Their haustoria, modified root structures, penetrating the host branch and extracting water and dissolved minerals from the host xylem while conducting their own photosynthesis. In tropical forests, the diversity and ecological importance of hemiparasitic plants is far greater than in temperate regions. Lorantheaceae mistletoes in African savanna woodlands can substantially reduce the productivity of their host trees while simultaneously providing critical food resources, fruits and nectar, for specialist bird species that in turn disperse their seeds. Full parasites, plants with no chlorophyll and no independent photosynthesis, also adopt tree-like growth forms in some cases. Arsuthobium, the dwarf mistletoes of North American conifers, cause witches' brooms, dense, abnormal shoot proliferations in their hosts, and are significant pathogens of commercial forestry, reducing timber quality and tree vigor across millions of hectares of productive forest. While the above-ground portions of trees attract most of our attention, the root systems responsible for anchoring, feeding, and, through their mycorrhizal partnerships, networking individual trees extend through the soil in forms of extraordinary reach and complexity. Some of the most extensive root systems known belong not to the trees we typically associate with large size but to species adapted to poor soils where extensive exploration is the primary survival strategy. The extensive lateral root systems of pines in dry sandy soils can extend 20 to 30 meters from the trunk base, spreading far beyond the drip line of the canopy in a flat, disc-like network that efficiently intercepts the sparse rainfall that infiltrates sandy substrate. Desert trees such as the mesquite of North American deserts and the acacia species of African savannas develop root systems that penetrate many tens of meters vertically to reach deep water tables. The mesquite root of over 50 meters depth from a prospecting mine shaft has entered botanical legend. The total root length of a single mature tree, including all its fine absorptive rootlets, reaches figures almost beyond intuitive comprehension. Estimates for large temperate trees commonly cite total root lengths of several hundred kilometers, and when the hyphae of associated mycorrhizal fungi are included, this extends to tens of thousands of kilometers. A three-dimensional exploration of the soil matrix of extraordinary extent. Beneath the feet of every person walking through a forest, a biological community of staggering diversity is engaged in chemistry whose complexity rivals anything happening in the canopy above. A single gram of healthy forest soil contains somewhere between 100 million and 1 billion bacterial cells representing thousands of species, along with comparable densities of archaea, protists, nematodes, and the omnipresent fungal hyphae that link everything together. This soil microbiome is not merely a passive decomposer of organic matter. It is an active mediator of nutrient availability to tree roots, a regulator of soil structure and water retention, a controller of plant pathogen populations, and a participant in the chemical signaling networks that influence tree physiology. Tree species actively shape the microbiome in their immediate rhizosphere, the narrow zone of soil immediately surrounding root surfaces, through the specific composition of the sugars, amino acids, and secondary metabolites they exude into the soil. Different tree species produce characteristically different rhizosphere microbiomes, and the composition of the bacterial community around a beech root is measurably distinct from that around an oak root growing in the same soil. These tree-specific microbiomes in turn influence nutrient cycling, decomposition rates, and the availability of plant-usable forms of nitrogen and phosphorus in ways that feed back to influence tree growth and health. The tree and its soil microbiome co-constitute one another in a relationship whose full complexity is only beginning to be mapped. Trees are profoundly responsive to the physical forces that act upon them throughout their lives, and among the most important of these is mechanical stress, the bending, swaying, and compression imposed by wind, snow, and the tree's own weight. The process by which plants and trees modify their growth and anatomy in response to mechanical stimulation is called thigmomorphogenesis, and its effects on tree form and wood quality are pervasive and consequential. Trees that experience regular wind stress grow shorter, stockier trunks with higher wood density and greater resistance to bending than trees of the same species grown in sheltered conditions, a response mediated by the hormone ethylene, released in greater quantities in tissues under mechanical stress. The wood produced on the compression side of a leaning trunk or a bent branch is anatomically distinct from normal wood, In conifers it is denser and contains more lignin, forming what would anatomists call compression wood. In broad-leaved trees, the opposite side of the bend produces tension wood, with a higher proportion of cellulose and different mechanical properties. These wood types represent active responses to mechanical loading. The tree is, in effect, continuously monitoring its own structural stresses and adjusting the properties of the wood it produces in order to resist them. Trees grown in greenhouses without exposure to wind consistently produce weaker wood and are more susceptible to wind damage when transplanted, a finding with practical implications for the production of trees for urban planting. The conversion of forests to other land uses, agriculture, pasture, infrastructure, urban development, is proceeding at a rate that constitutes one of the defining environmental crises of the contemporary era. Between 1990 and 2020, the world lost approximately 178 million hectares of forest, an area approximately the size of Libya. The pace of loss has slowed somewhat in some regions but accelerated in others, particularly in the Amazon, Congo, and Southeast Asian forests, which concentrate the greatest share of remaining tropical biodiversity and carbon. The consequences of deforestation cascade far beyond the forest edges. Disrupted rainfall patterns extend hundreds of kilometers beyond deforested areas. Increased sedimentation from exposed soils degrades rivers and coastal fisheries downstream. The extinction of forest-dependent species eliminates evolutionary lineages that required millions of years to produce. Perhaps the most counterintuitive consequence of large-scale tropical deforestation is its potential to accelerate rather than simply accompany climate change. As the trees that generate regional rainfall cycles are removed, rainfall decreases, remaining forests are stressed by drought, fire frequency increases, and the remaining forest fragments begin to release their carbon stores. The Amazon basin may already be approaching a tipping point at which deforestation, drought, and fire become self-reinforcing, a transition from the world's largest carbon sink to a carbon source of global significance. As climate change accelerates, the species compositions of forests around the world are beginning to shift in response to changing temperature and precipitation regimes, and foresters and ecologists are beginning to grapple with the question of how to manage this transition. In temperate regions, species currently limited to the southern or lower elevation portions of their ranges are expanding into previously unsuitable environments as those environments warm, while species adapted to cooler, more northerly conditions are retreating or declining at the southern edges of their ranges. In managed forests, this creates a dilemma. Trees planted today will still be growing in 50 or 100 years, when climatic conditions may be substantially different from those prevailing today. Planting species well adapted to current conditions may create forests poorly adapted to future conditions. Planting species pre-adapted to projected future conditions requires accepting significant uncertainty about how that future will unfold. Researchers in several countries are developing provenance trials, experimental plantings of the same species from multiple geographic origins, to identify seed sources whose genetic constitution pre-adapts them to warmer and drier conditions. Some ecologists argue for deliberately introducing tree species from more southerly regions, assisted migration, to maintain forest function under projected future climates. Others argue that managing forests for structural diversity and genetic diversity provides more robust resilience than selecting for specific projected conditions whose realization is uncertain. The question of what forests to grow for an uncertain future is among the most pressing in applied ecology. The material properties of wood, that cellular composite of lignin, cellulose, and hemicellulose produced by the cambium of trees, are so distinctive and so useful to human civilization that they have been studied, exploited, and celebrated across all of recorded history. Wood is simultaneously light and strong, flexible and hard, workable and durable, a combination of properties that no single synthetic material has convincingly replicated across all its dimensions. Its strength-to-weight ratio rivals steel in certain loading configurations. Its acoustic properties have made it the preferred material for stringed and percussion instruments for millennia. Its thermal insulation properties surpass most common building materials. Its capacity to be worked with simple tools and to be joined without adhesive or fastener through pegged, mortised, or dovetailed joints enabled the construction of complex structures without industrial technology. The specific anatomical structure of wood from different species produces materials of dramatically different character. The straight, coarse grain of ash is ideal for tool handles requiring impact resistance. The fine, even grain of boxwood for carving. The interlocked grain of elm for wheel hubs that must resist splitting. The flexible, long-fibered quality of Osage orange for bows. Traditional craftspeople who worked with specific wood species daily accumulated knowledge about the material properties of those species that has in many cases been formally validated by materials science only in recent decades. Another reminder of the depth of human understanding of trees accumulated through intimate, practical engagement. One of the most ecologically critical relationships in temperate forests is the timing between the opening of oak buds in spring and the hatching of the winter moth and other spring-feeding caterpillars whose populations are calibrated to feed on the flush of new, soft, barely defended oak leaves. This phenological synchrony, the matching of insect emergence to plant leaf flush, has been precisely co-tuned over evolutionary time because it is the primary determinant of caterpillar survival and ultimately of the breeding success of the blue tit, great tit, and pied flycatcher birds that depend on caterpillar abundance for feeding their nestlings. Climate change is disrupting this intricate three-way timing. Spring warming is advancing both bud burst and caterpillar hatching, but not at the same rate, creating mismatches between peak caterpillar availability and peak nestling food demand. Long-term studies in Dutch oak woodland, conducted by the ecologist Marcel Visser and colleagues over more than 40 years, have documented the progressive uncoupling of this timing relationship and its measurable consequences for bird breeding success. The oak tree sits at the center of this phenological web, its spring timing influencing not only caterpillar populations but the timing of leaf senescence, acorn production, and the autumn fattening of jays, wood pigeons, and deer. A cascade of temporal dependencies that is being disrupted at every link by the asymmetric responses of different species to warming temperatures. The behavior of trees, the direction of their growth, the timing of their dormancy and awakening, their responses to stress, their allocation of resources between competing demands, is orchestrated by a suite of chemical hormones produced in specific tissues and transported to their sites of action through xylem, phloem, and intercellular diffusion. The plant hormone system, while fundamentally different from the endocrine systems of animals in its mechanisms and molecular identities, achieves a comparable integration of information and coordination of response across a complex, multi-tissue organism. Auxin, produced primarily in the actively dividing cells of shoot tips, coordinates directional growth by promoting cell elongation on the shaded side of a shoot, causing it to bend toward the light, a phenomenon called phototropism. Gibberellins, produced in young leaves and seeds, promote cell elongation and stimulate germination and flowering. Cytokinins, produced in root tips, promote cell division and delay leaf senescence, traveling upward through the xylem to signal root activity to the shoot. Abscisic acid, produced in leaves under drought stress, causes stomata to close and promotes dormancy preparation. Ethylene, a gas produced in damaged, ripening, or senescent tissue, coordinates leaf drop, accelerates fruit ripening, and mediates responses to mechanical stress. These hormones interact in complex networks of promotion and suppression, and the integration of their signals represents the tree's primary mechanism for coordinating the activity of an organism that may be 100 meters tall yet lacks any central nervous system. Beyond their extraordinary carbon storage capacity and their role as coastal protection infrastructure, mangrove forests support some of the most productive marine fisheries in the tropical world, providing nursery habitat for the juvenile stages of a remarkable proportion of all commercially important tropical fish and shrimp species. The complex three-dimensional structure of mangrove root systems, the branching aerial prop roots of red mangroves, the tangled pneumatophores of black mangroves, creates shelter from predators that juvenile fish and crustaceans depend on during the critical early phases of their development. Nutrients leaching from decomposing mangrove leaf litter fuel a detritus-based food web of extraordinary productivity, supporting the invertebrate communities that constitute the primary food source for juvenile fish. Studies comparing fish catches adjacent to intact mangroves versus areas where mangroves have been cleared consistently find higher fish biomass, greater species diversity, and higher catch rates near intact forest. The fishers who depend on inshore tropical fisheries, many of them among the world's most economically vulnerable communities, are consequently among the groups most immediately harmed by mangrove destruction. The replacement of mangroves with shrimp aquaculture ponds, which has driven much of the mangrove loss of the past half-century, destroys the habitat that sustains the wild shrimp fisheries upon which neighboring communities have depended for generations, replacing a sustainable, self-renewing resource with an operation that typically exhausts its substrate within a decade. The gap between the way trees have been imagined in human mythology and the biological reality revealed by science has, over the past few decades, narrowed in ways that are genuinely remarkable. Many traditions imagine trees as conscious beings, as spirits with intentions, as channels between worlds, as organisms capable of relationship with humans in ways that transcended the merely material. Science has now confirmed that trees are far more behaviorally complex than the paradigm of the passive, unconscious plant ever allowed. They actively respond to their environment, communicate through chemical and fungal channels, appear to allocate resources in ways that favor related individuals, and maintain memory of past environmental stresses in their anatomy and physiology. This does not mean that trees are conscious in any way meaningfully analogous to animal consciousness. The question is genuinely open, and most plant biologists would vigorously resist that framing. What it does mean is that the intuition embedded in world mythologies, that trees are participants rather than mere backdrop, that they are in relationship with the living world around them rather than passive objects within it, has turned out to be, in significant respects, biologically warranted. The Shinto kami of the sacred tree, the Celtic dryad, the Norse Yggdrasil with its inhabited complexity, these imaginings projected onto trees qualities that science is now confirming in different, more precise, and ultimately more astonishing terms. The documented psychological and neurological effects of time spent in or near trees extend beyond the well-studied immune and stress hormone effects of Shinrin-yoku into the domain of attention, mood, and cognitive performance. Attention restoration theory, developed by environmental psychologists Rachel and Stephen Kaplan in the 1980s and subsequently supported by considerable empirical research, proposes that natural environments, and forested environments in particular, restore the directed attention capacities depleted by urban and work environments, providing what the Kaplans term fascination, the effortless, involuntary attention evoked by natural scenes, in place of the directed, effortful attention that modern cognitive life demands continuously. Functional neuroimaging studies have demonstrated that viewing natural scenes activates different patterns of neural activity than viewing urban scenes, with reduced activity in the subgenual prefrontal cortex, an area associated with rumination and negative self-referential thought. Controlled experimental studies consistently find improved performance on attention and creativity tests following exposure to natural settings. Hospital patients recovering from surgery whose rooms overlooked trees rather than a brick wall recovered faster, required less pain medication, and were discharged sooner. Children with attention deficit disorders show measurably reduced symptoms after time outdoors in natural settings compared to indoor or urban outdoor environments. Trees, it appears, are not merely metabolically and ecologically significant to human life. They are psychologically necessary in ways whose depth and complexity science is only beginning to measure. Among the most mysterious and visually arresting phenomena of the nocturnal forest is the bioluminescence of certain wood decay fungi, the cold, greenish light emitted by the mycelium and sometimes the fruiting bodies of species in the genera Mycena, Pinellus, and Omphalodus, among others. This phenomenon, known as foxfire or fairy fire, has been documented in forests worldwide and attributed in folklore to spirits, fairies, and the ghosts of trees. The biological mechanism of fungal bioluminescence was fully elucidated only in 2018, when Russian and Brazilian researchers identified the specific chemical pathway responsible, a luciferin compound derived from caffeic acid, itself derived from lignin. The wood polymer the fungi are decomposing is oxidized by a luciferase enzyme in the presence of oxygen, releasing light in the process. The evolutionary significance of bioluminescence in fungi remains genuinely uncertain. One hypothesis proposes that the light attracts insects at night that disperse fungal spores, providing a reproductive advantage. But the precise adaptive significance has not been definitively established. What is clear is that bioluminescent fungi are associated with specific stages of wood decay, and their light, persistent, cold, and unwavering, suffuses the forest floor in certain conditions with an illumination so alien and beautiful that it is comprehensible why every culture that encountered it attributed it to the supernatural. In this case, the reality is, arguably, stranger. For all the revelations of the past decades of tree science, the mycorrhizal networks, the chemical communication, the electrical signaling, the extraordinary longevity and ecological complexity, there remains a vast territory of tree biology that science has not yet adequately mapped. We do not fully understand how trees integrate information arriving simultaneously through multiple channels, electrical signals, hormonal gradients, light and temperature cues, chemical messages from fungal networks, into coherent, adaptive responses. We do not understand what determines the lifespan of trees, why some species live for hundreds of years and others for thousands, and what biological processes set those limits. The deep internal dynamics of mycorrhizal networks, which connections form, which dissolve, how the flow of resources through the network is regulated, remain poorly resolved. We do not know how many species of mycorrhizal fungi exist worldwide, or how their communities are structured, or how stable they are over time. The mechanisms of kin recognition in trees, if it genuinely occurs, are not identified. The molecular basis of how a tree ring forms, the precise cellular and hormonal sequence that produces the seasonal transition from spring to summer wood, is understood in outline but not in detail. The forest is, in a very real sense, still a frontier, a domain in which genuinely new discoveries remain available to anyone patient enough to ask the right questions. That frontier is one of the most intellectually exciting places in all of science. The planting of trees in memory of the dead is among the most ancient and geographically widespread of human commemorative practices, and its persistence across cultures separated by millennia and oceans suggests that it answers something deep in the human apprehension of death and continuity. The association of specific tree species with burial and the afterlife is ancient. Yews stood in Celtic burial grounds thousands of years before Christianity, their extraordinary longevity making them natural symbols of continuity between the living and the dead. The yew's habit of sending down roots from its lowest branches, creating new trunks that outlast the original, potentially extending the life of the individual tree for tens of thousands of years in theory, gave it a biological basis for its symbolic association with resurrection. Ancient Japanese traditions of planting trees over the graves of the dead have given way in contemporary Japan to practices of burying ashes beneath a young tree that will grow over the remains, creating a personal forest memorial that some see as both ecologically and spiritually preferable to the land use of conventional cemeteries. The concept of the memorial forest, where human remains are interred in degradable containers beneath a biodiverse wood rather than in a conventional cemetery, is gaining traction in Europe and North America as a response both to the land pressure of conventional burial and to the desire for a more ecologically integrated form of remembrance, one that returns the carbon and minerals of a human body to the living system of the forest. Looking forward from the present moment, from a world in which forests are under simultaneous pressure from climate change, deforestation, invasive species, and atmospheric pollution, toward the deep future of forest ecosystems requires an active imagination that must hold both profound uncertainty and profound possibility in suspension. The evolutionary resilience of trees is extraordinary. Forests have survived every mass extinction event, every ice age, every volcanic winter and meteor impact that Earth has experienced over the past 350 million years. They have done so through migration, adaptation, and the extraordinary reserve of genetic diversity maintained within widespread species populations. The current crisis, anthropogenic climate change at a pace unprecedented in the Cenozoic era, combined with habitat fragmentation that blocks the migration routes trees need to track shifting climates, is genuinely different in character from past disturbances. But it is not necessarily insurmountable. If the world's remaining primary forests are protected, allowing natural regeneration and migration, if managed forests are redesigned for structural diversity and mixed species composition, If the mycorrhizal networks that underpin forest function are recognized as critical infrastructure deserving as much protection as the trees they serve, and if the communities and cultures that have maintained forests for millennia are supported in continuing to do so, then the deep future of forests, though it will be different from their present, need not be one of impoverishment. Trees have always been longer-sighted than the creatures that live beneath them. We began this journey in the silence that is not silence, in the living hum of a forest that breathes, communicates, remembers, and endures across timescales that make the entirety of human history seem brief. We have traced the water climbing in defiance of gravity through vessels finer than a hair, followed the carbon as it moves from leaf to root to fungus and back, counted the years in cross-sections of wood that held them like the pages of a book written in growth rather than ink. We have stood in imagination before the oldest trees on Earth and tried to comprehend what 5,000 years of continuous existence means for an organism that experiences time so differently from us. We have descended into the mycorrhizal networks and witnessed the quiet commerce of the forest underground. We have parsed the chemical vocabulary of defense and invitation and warning drifting invisibly through forest air. We have found in the mythology of 100 cultures the flickering intuition of something that science is now illuminating from a different angle. Trees are the oldest, largest, and in many ways the most complex organisms on this planet. They built the soil we farm, the air we breathe, the rain that falls on our fields. They hold the climate stable, sustain the rivers, and support more life than any other structure on Earth. What they ask in return is remarkably little. Only that we slow down, pay attention, and perhaps remember, as our ancestors always knew, that we are not separate from the forest. We came from it. We belong to it still.